Okay, good evening, everyone, on this wonderful monsoon Saturday evening. So today we are here with the National Midterm CME 2022. This has been organized by the Indian Association of Biological Psychiatry in association with the Bombay Psychiatry Society. And it's going to be a two-day event. Today is going to be an online event for which we've come here today. And tomorrow will be an offline event, will be held in the holiday in Andheri. So without any further ado, let's start off. The theme for today's CME is applications of biological psychiatry in the clinical practice. So I would first like to invite Dr. Sunil Mittal to inaugurate the session.
Thank you. Thank you, Dr. Projector. And thank you, Dr. Karya. I see Dr. Shetty here, and also, of course, Dr. Rukseda for putting it all together. And it's a pleasure here to be this evening, like Projector said, on this monsoon evening. It's drizzling, just beginning to drizzle outside here in Delhi. So it's a pleasure being here to the initiation of this National Midterm CME 2022 of the Indian Association of Biological Psychiatry and the Bombay Psychiatric Society. So firstly, thanks BPS for supporting this activity and sort of anchoring it, hosting it there. The Indian, for people who may be new here, the Indian Association of Biological Psychiatry is an association committed to the biological aspect of psychiatry, the mental health aspect being biological there, etc., and the disorders, treatments. But then, of course, there may be many who feel that it's consciousness that pervades matter, and that's what came first. But then we cannot disagree that ultimately we are biological in substrate, and whatever happens, happens on this substrate. The biology that makes us all, that's what, as scientific people with the background of science, is what we would believe. Whatever it be, this division, of course, we know is this Cartesian division is not really true. But then we also understand that like today there was this study which was released about children who have had childhood abuse later in life developing schizophrenia. Now this was released last week. So we know this earlier if the observations were psychological in nature and it was psychological experiences that were spoken about. Today we understand the biological, the neurocorrelates and what happens inside the brain. And that's the advancement of science. And in keeping with Iabp, it brings you the latest what's happening on the frontiers of biological psychiatry. As you can see, look, if you look at the program for today evening and tomorrow, you'll see that the topics are really, if I can say, topical. They're today, they're happening today. The science is unfolding there. And as is the tradition, the scientific committee and the CME committee of the IABP have pulled in some of the best speakers on these topics that you can have. So it's a privilege for anybody to be attending this CME. Today's session, today's evening is devoted to the young psychiatrists and the PGs. And that's why if you have a look at the topics that are there, the whole question of the, like I was saying, the recent advances in the biology of schizophrenia and the sort of study I alluded to, but of course there'll be a huge ground covered in this. Then neuromodulation techniques and post-ECT, other neuromodulation techniques have come in, whether it's Tdcs, CES, or of course you have on the seizure side, the invasive magnetic seizure therapy or the deep brain stimulation. But then you also have transcranial magnetic stimulation and the deep TMS, which is here. So neuromodulation techniques, and again, we have an eminent speaker there. Then, of course, the conundrum that has been epilepsy and psychiatry. You know, I was speaking to an epileptologist, and he says he's a professor at AIIMS, one of the most reputed on the the International League Against Epilepsy, represents this part of the world, Asia on it. And he said, Dr. Jain, he said, And actually, Sunil, epilepsy is something you guys should be treating. It's episodic. It occurs for a short while. And then, of course, the medicines that you use, which you call anti-epileptics, I can call them mood stabilizers, and I use them to treat epilepsy. So it could be either way to look at it. So today we have this epilepsy and psychiatry, what PGs should know. I'm going to end here. Welcome to the CME. And let's go without further ado. Let's just move with the scientific program. And back to you, Project.
Thank you, sir. Thank you. Now I would like to call in Dr. Rukshida Saida, who is the secretary of the Iabp, just for a few words. Ma'am.
Thank you, Dr. Prajakta. At the outset, I'm grateful to Dr. Sunil Mittal, sir. He's a senior psychiatrist from Delhi, but more especially, he is the senior vice president of Indian Association of Biological Psychiatry. He was very kind to take time out to introduce us to not only just the midterm CME, but also to IABP. And his words of encouragement and wisdom are going to set the stage for the coming today and tomorrow's event. This event is especially dear to me because as a general secretary, this is the first midterm CME that we haven't had in a lot of time. And we are also going to have an in-person physical conference tomorrow, which again is going to be the sole physical conference of IABP in my tenure at this since the last couple of years. So clearly, I'm really excited and I'm really grateful to all the delegates and the participants who are listening in and who are eager to learn and learn more about virology. We wanted to dedicate this first session today of Saturday more towards the PGTs and young psychiatrists, and also to the oldies like me who want to revise a few topics. Those of us who are not as well read as Dr. Sunil, you can definitely learn a few new tricks as well. I take this opportunity to thank the local organizing team with Dr. Nalesha, Dr. Avinash D'Souza, and Dr. Sagar Karya and his team. And also a special thanks to Dr. Bombay Psychiatric Society, where Dr. Prajakta and Dr. Karya are on the EC. Dr. Kavan Lakrawala, who's the president, has been very supportive and given a blanket say yes, that Rukshida, whatever you want is okay for us. So that's been very, very heartening because at heart one always belongs to BPS, even if you've served only for a few years. It's an honor that all the speakers that we approached, nobody hesitated and everybody agreed and everybody said yes for the sessions. So we're very obliged to them, and our chairpersons have taken time out from their sessions as well. We at IABP are not only just diligent about time, but we are very particular about the content of our presentations, the speakers, and also the chairs who are well suited to address that session and modulate that session. So because we know that apart from presentation, Q&A is where we learn a lot. So at this point, I'm going to hand over to Dr. Prajakta for further sessions, and hopefully we are looking forward to a day and a half of learning, and best of luck to all of us. Thank you, Prajakta.
Thank you, ma'am. So like ma'am and sir have already said that this session is mostly for the postgraduate students, and we have 3 wonderful lectures lined up. So the first session is going to be on recent advances in biology of psychiatry by Dr. Suhas Satish. For this session, we have 2 chairpersons. The first being Dr. Jyoti Shetty. She is the professor and head Department of Psychiatry in Bharati Vidyapeeth Medical College, Pune. She has been a member of the Institutional Ethics Committee at Bharati Vidyapeeth. She has been the convener of Child Psychiatry Subcommittee, the convener of Forensic Psychiatry and Law Subspecialty. Last year, she was the co-chairperson of the IPS Perinatal Psychiatry Task Force and the co-chairperson of the IPS West Zone UG Education Committee. This year, she's the advisor at the IPS POXO and JJ Act Task Force. Ma'am has been a past IPS Pune branch president and the president of the PPA as well. Ma'am is a wonderful teacher. I've been her UG student in Bharati Vidyapeeth. So welcome, ma'am.
Thank you.
Our second chairperson is Dr. Ashwin Mohan. Sir is an MD from the prestigious PGI. He also has a postgraduate diploma in medical law and ethics from National Law School in Bangalore. Sir is a senior practicing consultant at Chandigarh. His areas of interest include substance abuse disorder, mental health policies, law and advocacy. Sir is currently the chair of the IPS Addictive Disorder Specialty Section and the EC member of the Iabp EC member of Addiction Psychiatry Society of India. Welcome, sir.
Thank you so much. Thank you.
If you could introduce our speaker, one of the chairpersons.
So, Dr. Jyoti,
yeah,
you can do the honors.
Yes. Thank you. It gives me great pleasure. Thank you at the outset, IABP, for inviting me to chair this session. It gives me great pleasure to introduce today's speaker, Dr. Suhas Satish, who's the Assistant Professor of Psychiatry at NIMHANS. He's an MBBS from the MS Ramaiah Medical College, Bangalore, and he's an MD Psychiatry from NIMHANS. He's done a fellowship in clinical neurosciences and therapeutics in schizophrenia from NIMHANS, and his research interests remain schizophrenia, neuromodulation, community psychiatry, telemedicine, psychopharmacology, and leadership. He has several publications. He has also received lots of awards and honors, like the WPA 19 WPA Congress of Psychiatry Early Career Fellowship Award in 2019, the Must Enrich Grant Award recipient from the Indian Teachers of Psychiatry, the ITOPS initiative in 2020-22, Community Psychiatry Specialty Section IPS Shima Trust, Karnataka, co-recipient of the third prize in the Best Community Psychiatry Research Paper in 2021. He's also received the IPS South Zone, co-recipient of the Dr. S.S. Jairam Award for Best Paper in 2021. He's also received the first prize in the paper poster presentation in the 7th Leadership and Professional Skills course in earlier. Career psychiatrist at Indira Gandhi National Graduate Diploma in 2015, and he's for best outgoing student.
I think we've lost Dr. Jyoti. She's logging in from a cell. So maybe this is the time I take over from.
Yeah, thank you. I think I got locked out.
Yeah. No,
yeah.
So
sorry.
Definitely welcoming Dr. Suhas Satish, and very accomplished psychiatrist and assistant professor of psychiatry from NIMHANS. I'd also like to thank IABP and Bombay Psychiatric Society and all the organizers for inviting me as a chair here. So since we're already a bit late, about 10 minutes late, I'll invite Dr. Suhas to begin his talk. Thank you.
Thank you, sir. Thank you, ma'am. I'm going to start sharing my screen now. At the outset, I would like to thank the IABP and the Bombay Psychiatry Society for inviting me to deliver this talk for psychiatry postgraduates, which is such an important talk when we look at the postgraduate learning perspectives to understand the neurobiology of psychiatric disorders. Especially, I've been asked to speak about the recent advances in the neurobiology of schizophrenia. So when we speak about the biology of schizophrenia, we of course have to look back from the womb to the development of schizophrenia clinical phenotype. And then when we speak about all the changes that happen therein, we speak about different factors that contribute to schizophrenia, one among which is the genetics. In genetics, of course, we have had fairly modest amount of success in determining the biological reasons for schizophrenia. Some major genes that have been implicated include dysbindin, neuregulin, the disrupted in schizophrenia genes, and the association studies have also led to the identification of few other implicated genes. But apart from the genetic studies, there have also been substantial studies that have looked into the neurotransmitter systems in schizophrenia. And there have been imaging studies which promised a lot in the early 1970s and 80s, and which have gone on to definitely add to our understanding, but not as much as we had hoped. And of course, there is the clinical-based understanding and the applications in the biology of schizophrenia. Keeping in mind that this is designed to refresh, update, and help psychiatry postgraduates, I'm going to do a bit of a back to the future kind of a talk in which I have to introduce a few concepts before I talk about what is fairly recent in the field of schizophrenia. When you speak about the updates in the genetics of schizophrenia, apart from the genes that I had mentioned in my previous slide, the Schema Consortium has actually identified that although there are about more than 6,671 genes that are differentially expressed in schizophrenia across various studies that have been reported worldwide, only about 10 ultra-rare coding variants actually provide substantial risk towards the development of schizophrenia. Therefore, it is fairly common nowadays for psychiatric postgraduates and all of us who are working in the field of mental health to understand what is called as a polygenic risk score, because what is ultra-rare cannot actually be common in the clinical setting. And so we understand the common gene and the common variant and the pooled risk hypothesis, which says that we generally look at the expression of the common variants and the confer risk to the combination of all this risk. And that is the polygenic risk profile. But however, the Schema Consortium has recently, as past month, told us that the risk conferred by the polygenic risk profile accounts for about 20 to 24% of the heritability. Although we know that schizophrenia is a highly heritable disorder with 80% heritability, the polygenic risk profile only contributes to about 20 to 24% of the clinical variance in the phenotype. Now, when we talk about the neurocircuitry, we all know that schizophrenia is a disorder that is associated with the aberrations in different neurocircuits. And starting from top down, we know about the dorsolateral prefrontal cortex and the value that it adds towards mediating the action control and the top-down regulation, the thalamus and its role in internal representation, the ventromedial prefrontal cortex and its role in mediating the action selection based on the previous choice and analyzing the post-choice values, the anterior cingulate cortex, which tracks the progression of goal-directed sequences as to whether it is happening correctly or not. And of course, we have the caudate and the dopamine circuits, which I will be covering it in fair bit of detail. So let us address these neurotransmitter systems one by one. It was serendipitously discovered that there are multiple neurotransmitters that can be implicated in schizophrenia, ranging from dopamine, which is of course the favorite culprit. And after dopamine, there have also been other neurotransmitter systems, especially glutamate, which have then added substantially to understanding the neurobiology of schizophrenia. But however, when we look at all these neurotransmitter systems, we actually understand that in schizophrenia, that there is a central role for dopamine and glutamate. So just to refresh ourselves about the dopamine neurotransmitter systems, we know that there are 4 major dopamine pathways in the brain. That is the mesolimbic pathway, the mesocortical pathway, the nigrostriatal pathway, and the tuberoinfundibular pathway. But of all these things, we now understand that it is a hyperactivity at the mesolimbic pathway which produces the positive symptoms of schizophrenia, and hypoactivity at the mesocortical pathway which produces the negative symptoms of schizophrenia. Fairly simplistic to follow, and this is what we base our knowledge on, but we'll build up from here. So the dopamine hypothesis of schizophrenia goes on to say that the dopamine blockade, which has led to an improvement in the schizophrenia, probably alludes to the hyperdopaminergic state that is present in the brain, reason as the responsible for the expression of the positive psychotic symptoms. And therefore, one of the earliest theories that came about is the fact that, okay, there is some amount of hyperdopaminergy that is happening in the brain, and that's leading towards the manifestation of psychosis. Then came the refined versions of these hypotheses. So then people realized that what Seaman and had published in 1975 as a hyperdopaminergic state is simplistic in its understanding because it doesn't explain the other symptom domains. And therefore, they talked about the hyperdopaminergy at the mesocortical circuits being the determinant of the negative and the cognitive symptom profile. Then came a very landmark article which spoke about the centrality of dopamine in a different way, in which they spoke about how dopamine is the wind of the psychotic fire and it might not actually be the determinant of the origin of the psychotic fire in schizophrenia. Multiple refinements of the dopamine hypothesis have now led to an understanding that it is perhaps the associative striatum which integrates the inputs from the prefrontal cortex and the thalamus, which in turn is mediated by the emotional processing and the perception from the amygdala and the hippocampus, and the bidirectional relationship between the associative striatum and the thalamus that leads to the manifestation and the expression of the psychotic symptoms. So the role of dopamine is actually fairly well established when we look at the associative striatal dysfunction in the manifestation of positive psychotic symptoms. However, it is not so clear when we look at the other symptom domains of schizophrenia, especially moving on towards the treatment refractory.
Give me 10 minutes, I'll come.
Yeah. So moving ahead, we need to understand couple of more important pathways in the neurobiology of schizophrenia. One is the reward pathway, which we all know is the pathway that involves the ventral tegmental area, the nucleus accumbens, and the prefrontal cortex. And I'll come to as to why I'm speaking about the reward pathway in schizophrenia in a bit. We all know that this is not a program on addiction, but I'll come back to it in a bit. So refining all our knowledge and understanding out of the dopamine and the role it plays in schizophrenia leads us to understanding that dopamine is actually in its centrality a mediator of pleasure. So let's say that there is an event X. That event X might be different for different people. It is a pleasurable event and it leads to a release of dopamine in the reward pathway and the experience of pleasure as a result of the same. As I said, that might be commonly known as food, sex, and water, because which are the biological determinants of pleasure, and therefore dopamine was involved centrally as a mediator of pleasure. Then came the knowledge derived from the understanding of the role of dopamine in substance use disorders, and then came the refined version, which is very useful in understanding schizophrenia, which is the fact that dopamine is actually a very important neurochemical which is implicated in predicting reward and mediating pleasure thereof. So the dopamine release actually precedes the pleasurable event, which is the anticipatory phase of the experience. So there is an expectation of a reward, so which releases, say, X amount of dopamine, and there is an actual event which releases a Y amount of dopamine. Now, if the expectation supersedes the actual event, then it leads to dysphoria, because I expected something to happen, but it did not happen. Whereas if the expectation is actually subservient to the actual event in the release of dopamine, it leads to pleasure, because all is well, I expected this to happen, and it was much more better than that. This particular hypothesis actually was not very good in explaining how aversive events can actually mediate behavior in schizophrenia. But later on, we came to know that dopamine is involved in aversive experiences as well, and the antipsychotic drugs, therefore, when they block the wanting but not the liking of an experience, can have a complex role to play in the manifestation of secondary negative symptoms. Continuing on with the third generation of dopamine and its role in schizophrenia came the neuroleptic induced supersensitivity psychosis theories given by Chuina. Now, in this, they spoke about how that there are actually not one but two varieties of schizophrenia, that is the hyperdopaminergic and the normodopaminergic schizophrenia. And they spoke about the dopamine supersensitivity. If it is there before antipsychotic treatment, there is a release of dopamine and it leads to a normal dopaminergic activity. Whereas in treatment-resistant schizophrenia, if it is a normal dopaminergic state and there is a release of dopamine, then there is decrease in the treatment response because there is no hyperdopaminergia that we are able to target in the first place. So therefore, they found out this as the reason why some people respond very well to antipsychotic medications and some people do not respond very well to antipsychotic medications. Then came the concept of dopamine supersensitivity, wherein there is an upregulation of the postsynaptic dopamine receptors, and therefore a person will actually have a new onset manifestation of say, motor manifestations in the form of tardive or abnormal movements, and a sudden increase in the manifestation of psychosis despite being stable on antipsychotic medications for quite some time. This is explained by the dopamine supersensitivity per se. So now we have discussed dopamine version 1, dopamine version 2, and dopamine version 3. Now I want to introduce you towards two other important networks, which is integral to the amalgamation of what I'm going to talk in the coming slides. The Salience Network, as we all know it, involves the dorsal ACC, the anterior insular thalamus, and the amygdala hippocampus pathway, along with the nucleus accumbens and the ventral tegmental area. We all know that there are two important networks that have been implicated in thinking and behavior. That is the default mode network and the central executive network. When we talk about the default mode network, it is the mode that is running by default in our brain at all periods of time, without any particular input, without any particular conscious kind of an effort or a process involved. And we know that there is a central executive network, which is determined by a conscious process, which is actually top-down in nature, which is determined by the person himself. And then there is an anti-correlation of the neurological areas and the brain areas that are implicated in these two networks, the default mode network and the central executive network. And who determines what state that the brain is to be in? And that's the Salience Network, which we introduced a slide back. So the Salience Network is very crucial in the detection and the integration of the emotional and the sensory stimulus, and it modulates the switch between an internally directed cognition of the default mode and an externally directed cognition of the central executive network. So let's actually bring these things together and how a salience hypothesis of dopamine comes together in manifesting psychosis. So let's say that there is an external event X and there is no abnormal salience and the dopamine is not released. The brain interprets it as a neutral event. Nothing special about this external event X. Now let's talk about an external event X. Then there is a salience that is associated with abnormal dopamine processing. Then it means to say that this event is given a very special significance. So that is when the brain or the body perceives it either as an attractive event or threatful, as an aversive event. And it leads to an amalgamation of conscious and subconscious attention and top-down mediated processing of what should be the goal towards or away from this attraction or aversive event, and a behavior which is manifest in the form of action taken to either go towards the pleasure or away from the threat that the brain is perceiving. So the external stimulus, therefore, leads to action based on the salience hypothesis and the role that it plays in determining the response to this external stimulus. Mind you, that the stimulus remains the same. The stimulus is not changing in both these scenarios. What is changing is the brain's processing of the hedonic pleasure associated with the stimulus, the reward prediction associated with the stimulus, and based on the prior learning mechanisms as to how much the particular stimulus is either a threat or a pleasure in mediating the action. A neutral stimulus results in no action, whereas an attractive or an aversive stimulus results in action. So in normal individuals, dopamine mediates the salience attribution to a neutral event in which it is actually kind of telling as to when the switch should not go on and off unnecessarily. Whereas in aberrant salience, which happens in the case of schizophrenia, what happens is that it leads to an action based on a neutral event wherein there should have been no action in the first place. Therefore, a whole lot of environmental stimulus that shouldn't actually have provoked a response from an individual starts provoking a response. For example, at the beginning of my talk, just because somebody coughed, that doesn't mean that it is important or I should be giving a lot of importance to it. But however, if there was an aberrant salience in my brain systems, I could probably interpret as somebody coughing just to ensure that my concentration is disturbed and so that they actually want to sabotage my presentation. That happens in the cases of schizophrenia. Having established these important processes, I want to now connect it to the two important phenotypes of schizophrenia, especially the positive syndrome phenotypes, that is the delusions and the hallucinations. In cases of delusions, most often than not, it is a neutral external stimulus which actually manifests as a delusion. And in case of hallucination, it is the abnormal internal representation, which is given a whole lot of undue focus and attention, which manifests as a hallucination. More of that down the line. So we have established that, there is aberrant dopamine release, and there is a formation of some sort of a salience to a neutral stimuli, and there's a persistence of the particular salience to the, despite of the absence of a stimuli. It shouldn't have persisted, but it is persisting because the salience is aberrant in nature. And then there is an altered kind of an experience of the world, leading to heightened senses and the manifestation of psychosis following the prodrome. So when we look at the manifestation of psychosis, there is and this beautifully captures what Mittal sir was talking about earlier, wherein he spoke about the amalgamation of biology with the thinking and the psychological processes, wherein there is a top-down cognitive explanation to these experiences, which lead to jumping to conclusion biases, altered attribution styles, and magical ideation, which leads to the formation of delusions. Similarly, there have been recent advances in the field of the dopamine and its role in schizophrenia to explain the negative symptoms as well, wherein they talk about the dysfunctional motivational salience circuit mediating the role in the negative syndrome of schizophrenia. There is a caveat about dopamine syndrome and the dopamine and its role in schizophrenia, because predominantly it explains delusions to a large extent. Okay, if there is salience, there is there is a whole lot of external stimulus that is coming in, and I'm developing all these new ideas if I'm having aberrant salience. But it doesn't explain hallucinations so much. More so, it doesn't explain the development of a sudden delusional idea, and how in a case of substance-induced psychosis, so there is rapid alterations from a state of psychosis to a state of normalcy. Therefore, we know that dopamine in itself is not essential, or neither is it sufficient to explain the neurobiology of schizophrenia. That's when came the role of other neurotransmitters, and we now know fairly well that glutamate plays a very critical role in the generation of positive symptoms of schizophrenia and also the negative symptoms of schizophrenia. It is proved beyond doubt that the NMDA receptor hyperfunction hypothesis and the positive symptoms of schizophrenia and the downstream effects on cognition are important, and therefore therapeutic targets have now started to emerge, specifically targeting this particular neurotransmitter system. It is of interest to note recent schizophrenia research publication in the prodrome points towards a better role for agents that are targeting NMDA receptors and glutamate rather than dopamine in prevention to the development of psychosis in high-risk individuals. As I pointed out, glutamate is not just implicated in the positive symptoms, but it is also implicated in thinking disturbances and the generation of a formal thought disorder due to the synaptic effects and the downstream effects that the glutamate has. GABA is a neurotransmitter that has also been implicated in schizophrenia, but however, it is more implicated in the cognitive deficits and the negative symptoms, and it in itself cannot explain the positive symptoms apart from its role as an intermediary circuit. All right, now that we have understood all that, let us go towards integrating these things more towards our clinical gains. So we now know that when we talk about perception, we either talk about the bottom-up processing, wherein there is a stimulus, and then from the stimulus, there is a perception of a stimulus, and then the processing of the stimulus, or there is a top-down processing that is involved. If I ask all of you to actually look at the particular figure, although there is only three structures which are present, none of which actually are there to complete the triangle. But our mind actually can place two triangles in this particular figure because of our experience of the priors. And what is this role that the priors play? I'll be again talking about in the subsequent slides. The Kunesa triangle experiment is very clear in showing us that whereas in part figure A, which is there towards the left of your screen, we are able to clearly place a triangle where it is not there. In figure B, we are unable to do so. Although it is of critical importance to note that both of them do not have a triangle in it. So we now understand that it is in an evolutionary sense an important role that the brain plays in as a necessity to make sense of our environment. And therefore, this sense can be made based on our prior experiences. All right. So we now know, okay, let me now integrate perception and move beyond the theory of perception as just a sensation. We now know that there is a sensory input, and that is actually related to the thalamus as a sensation. But perception is much higher than that, wherein it integrates the sense that it is deriving from this sensation, and it liaises with the prefrontal cortex and tries to make sense of this particular sensation. And recent neurobiological theories have actually talked about the synchronization of the gamma oscillations in thalamocortical networks and the role of the attention and working memory, which are critical towards maintaining a healthy sense of this perception. It is very important what I'm talking about, because in the subsequent talks, when Professor Shyam Sundar is going to talk about the newer neuromodulatory agents in schizophrenia, he's going to talk about how oscillations can be targeted and their importance in schizophrenia as well. So we now know that there is a source of a stimulus, there is an insight related to that source of a stimulus, and there is a placement of the stimulus in our brain, whether it is internal or external, and an amalgamation of all this information leads to a healthy sense of perception. When we started understanding the neurobiology of perception, and it gave us clues towards the understanding of the neurobiology of hallucinations per se. We first understood it in terms of the sensory deprivation or loss studies. Then we understood, okay, what are all the non-psychotic conditions in which hallucinations emerge? And we know that the panoramic hallucinations of epilepsy and the Charles Bonnet syndrome, which has pseudo hallucinations in nature, give us some clues that, okay, it is not a simple sensation-derived mediated psychopathology, but it is much higher than that. Subsequently came the theories based on the neuroimaging studies and the EEG studies, which gave us much more substantial understanding of the neurobiology of hallucinations. Integrating the neuroimaging studies and the EEG studies, we are fairly clear that the healthy oscillations in the human brain and the role of the gamma oscillations in the thalamocortical networks and the role it plays in mediating the salience to the perception, and the importance of recognizing the perception as coming from within or without, and putting the source as coming from within or without, and having the insight of understanding whether it is important or not, is very critical towards the whole world experience of the perception that we have. The earlier theories of the disinhibition model given by Hughlin-Jackson, the dissociation model given by Marazzi, and the perceptual release theory given by West have since been discarded to give an integrative theory, which is a cognitive perceptual theory, which amalgamates both the bottom-up and the top-down approaches. Then came a very, very important landmark theory in 2007, which speaks about the self-generated speech and the corollary discharge. All of us now know that we cannot tickle ourselves, but when we try to understand as to what is the reason that we cannot tickle ourselves, it is fairly kind of intriguing. But neurobiologically speaking, we now know that when there is a conscious mediation of a particular action by the brain, there is a prior copy of that particular action or a forward model that dampens the responsiveness to this particular stimuli that will be generated as a result of the action that I'm doing. This particular theory gone on to explain to us that if it is aberrant in the field of internally generated speech, then it can lead to what is called as an auditory hallucinations because of an aberrance between the healthy corollary discharge and the two-way communication that happens between the self-generated activity and the corollary discharge related activity. So we now know that there is a lot of proof that corollary discharge hypothesis has kind of obtained in the form of subthreshold vocal cord oscillations that happen with hallucinations. And we also now know that this is being targeted as an important area of intervention in schizophrenia in the form of Tdcs, which will be spoken about by my next speaker. Then there was also the misattribution model, which has been intricately kind of connected to the corollary discharge model to generate what is called as the amalgamated model, in which there is a generation of an internally misrepresentation of an internally generated stimulus, which comes as a hallucination. And then when this particular processes over a period of time coincide with an aberrant pruning that happens during the neurodevelopmental stages, and especially during adolescence, it leads to an pathoplasticity and then maintenance of this abnormal kind of an experience. This, when it is combined with the neurological cognitive processing deficits that happen with schizophrenia, leads more and more towards the generation of a network of abnormal inferences and the delusions, which maintains this perpetual state of psychosis. So we've spoken about this. So we now know that the comprehensive model of hallucinations now combines all the theories that have actually kind of been declared until now, wherein it says that, okay, there is a release phenomenon which can explain only some states of hallucinations. Then there is some irritative processes that are involved, which can again explain organic psychosis. The attentional spotlight model can explain some sort of a generation of hallucinatory experience, but it's really the cognitive root and the coronary discharge hypothesis that explains the generation and the maintenance of the hallucinatory phenomena. So that has been actually summarized in this particular slide. Since I have actually spoken about this particular slide, I'm going to skip it. So now let's understand a bit about how this comes together in case of delusions and understanding delusions. And a very important thing when it comes to understanding delusions is predictive coding. Predictive coding is something that our brain actually does by default from a very young age, wherein it tries to understand everything that happens in our world based on what has happened to it before that particular stimulus is there. If I ask all of you to read this sentence quickly and loudly, all of us would probably read, Jack and Jill went up the hill. It's fairly straightforward, right? Now, if I ask you to do the same thing once again and ask you to read this out loudly, all of you would say that, yeah, it's fairly clear. It's written that the last event was cancelled. What's so different about this? But what's very intriguing is these two apparently unconnected sentences had one thing in common, and that was the fact that in the first sentence and in the second sentence, the letters that were written in the graphical manner that it were written could actually mean either event or went, depending upon what you were reading. But your brain did not recognize this until this point of time, because there was a predictive prior. We all had learned the rhymes, the Jack and Jill rhymes when we were children, and we all actually are used to this particular sentence in conferences that the last event was cancelled. So therefore, even before your brain could actually make sense of the sensation and give the perception the true quality based on what it really could be, it went on to fill the meaning of that particular sentence based on predictive priors. Having established this, I can give you one more example of how predictive priors plays a role in our understanding of the world and how it relates to schizophrenia. If I ask all of you to actually kind of tell which among these two shapes would you name as Kiki, and which among these two shapes would you name as Booba? And I'm sure that most of us, if not all, would call the shape on the left, which is fairly star-shaped, as Kiki, and the shape on the right, which is kind of like a very loosely shaped structure, as Booba. Why is that? That is because of predictive priors. Kiki sounds like kinked and sharp, and the particular shape on the left has sharp edges, and Booba sounds like rubber and flubber or blubber, and that loosely kind of shaped objects on the right, or irregularly shaped object on the right, actually kind of is named by Booba. And why is that? Why is that all of us actually think in the same direction? That is again because of the predictive priors. Now, after having explained about predictive priors, I want to now talk about three important pieces of information that is important for the brain to understand the world for what it really is. One is a template matching model. I can understand what I'm seeing, what I'm perceiving, what I'm actually kind of feeling based on the templates that I've had before. If I put a foreign script in front of you right now, none of us, including me, will be able to read it because it doesn't match the template of anything that I've known before. Then there is a feature matching model, wherein I understand the shapes of objects, the pattern that is there in the things that is being presented to me. And there is a configure model wherein I try to understand it in terms of the holistic experience that I'm having of this particular perception. Now, if you combine the theory of perception given by Helmholtz and the corollary discharge and the theory of delusion that I actually kind of spoke about just now, there are several unconscious processes that are there that interprets a current stimulus on the basis of a prior experience, and that interpretation is the perception, and any abnormalities in this interpretative process will lead to the dual presentation of a delusion and the hallucination, because the brain is actively trying to predict the present information or the present environment based on prior experience, intention, and a goal. And when that particular process becomes abnormal or skewed, that leads to the development of the schizophrenia positive syndrome. So we actually, our brain is actually more Bayesian than we think, and it operates through the predictive coding mechanisms that I have described. And therefore, this plays a crucial role in the neurobiology of delusions and the maintenance of the delusional processes. So we now know that over a period of time, remember my previous slides wherein I spoke about X being greater than Y and Y being greater than X. So if the prediction actually supersedes the reward, then it leads to a constant states of dysphoria and an increasing sense of uncomfortableness in the human brain, where it says that I have to make sense. I mean, there are so many uncanny coincidences that are happening over here. And so my senses become sharpened. I start to begin this particular, there is a new world being formed. There is an epiphany. And over a period of time, there is a new world with a new being, a new meaning attached to everything that happens around me that leads to the tipping point and manifestation of the psychosis per se. And over a period of time, there is a dual action between this particular predictive errors and the aberrant corollary discharges in maintaining the delusion and the hallucinatory experiences. So briefly touching upon the neurobiology of resistant schizophrenia, we now know that perhaps in resistant schizophrenia, it does not have a more unique neurobiology or a separate neurobiology per se, but we now know that it is perhaps more manifestation of a schizophrenia that has manifested earlier, or it is associated with more genetic loading or environmental deficits, and it is leading to more poorer outcomes and poorer treatment responsiveness. But it is associated with definite neuroimaging correlates, which actually kind of point towards some clues on picking up whether the person can be resistant or not. Mind you, none of which have clinical implications until now, but it is only at a level wherein we can retrospectively examine whether they were associated with treatment resistance or not. Regarding the genetics of resistant states also, that there are some indirect clues that give us a predisposition towards supersensitivity psychosis and some copy number variants that are associated with resistant schizophrenia. But however, it is fairly clear that risk score in the percentage, it directly doesn't correlate with the manifestation of a resistant schizophrenia. So there are several inconsistencies, and therefore we are not at a stage wherein we can predict somebody might develop resistant schizophrenia or not. Coming to some of the other neurobiological developments, we now know that the inflammatory hypothesis and the stress and the bidirectional relationship that it shares with the thinking and the behavior patterns have actually kind of shown that it could be associated with the maintenance and the worsening of psychosis. And indirectly, this is also alluded to by the connection that it has with the gut-brain axis. Again, these are at an investigational stage, and it doesn't have any translational implications. So just to summarize, we now know that the biological changes in resistant schizophrenia are still inconsistent and needs a lot of focus. And to understand as to why we are doing all this, why are we learning so much about, neurobiology of delusions and hallucinations? Does it really matter? At the end of the day, we are treating them with either second generation antipsychotics or augmenting them with some first generation antipsychotics, or giving some LAIs, or giving some neuromodulation. I will give you one small example of how our enhanced understanding of the neurobiology of schizophrenia has led to this development of this unique intervention, which is showing some promise. And that is the weak intensity neuromodulation of the cortical areas based on our understanding of neurobiology. Now, if we look at the neurobiology of schizophrenia, we now know that there is the temporoparietal and dorsolateral prefrontal cortical aberrations that can be actually corrected with weak intensity neuromodulation. When we talk about the temporoparietal junction, there is an hyperactivity with a significantly increased glutamate level. And when we talk about the dorsolateral prefrontal cortical dysfunction, there is a hyperactivity in schizophrenia with a decreased N-acetyl aspartate level, which has been identified. Now, can you think of a way in which we can suppress the temporoparietal junction and activate the dorsolateral prefrontal cortex? And would it really lead to an improvement in psychosis? This was some of the thinking that led to the development of, say, the earlier versions of the neuromodulation, which is the transcranial direct current stimulation, in which a cathedral stimulation to the TPJ and an anodal stimulation to the dorsolateral prefrontal cortex led to the attenuation of the psychosis, especially the hallucinatory experiences. And this is just one minor example of why we should be relentless in our pursuit of the neurobiology, keeping in mind that eventually it might lead to better treatment, better prognostication, and better outcomes for our patients. On that note, I would like to end. And I would like to once again thank the organizers, that is IABP and the Bombay Psychiatric Society, for giving me an opportunity to present this in front of young postgraduates, hopefully who would have found this presentation interesting, and hopefully it will stimulate them to read more and contribute more to this field. Thank you. Over to the chairperson.
Thank you so much, Dr. Suhas, for a very lucid and comprehensive presentation. You had this unenvious job of trying to decide about informing us about the recent biological advances in schizophrenia. It's actually very, very difficult to choose a starting point and decide from where do we advance and where do we go to. Plus, again, a difficult job of what to include and what to exclude, because this is a vast area of research, and I think 1 hour does no justice to this topic at all. So thank you for informing us. I think you chose some of the very commonest issues of hallucination and delusions, rather than focusing on other more complex topics where probably we need more time to really discuss. So thank you so much for simplifying for us. I mean, for whatever we can understand, despite all the difficulties. So thank you so much. I just have one question, and then I'll hand over to Dr. Jyoti too. People say there are no schizophrenias. I mean, there is no schizophrenia. There are schizophrenias, or the term itself should be abandoned. What is your take on that?
I think I fully agree with you, sir, on that. That is because Kraepelinian construct of schizophrenia is perhaps a prime example of structures sometimes becoming shackles, in the sense that we always try to explain the construct of schizophrenia based on the Kraepelinian dichotomy, and then on the overemphasis on the first rank symptoms of schizophrenia. And perhaps we have neglected Kleist and Leonard for far too long now. And now we realize that no two schizophrenias are actually alike, and many schizophrenia that we actually see in our clinical practice. Some of them are associated with affective syndrome, some of them with obsessions and compulsions, and some of them with the positive phenotype, some of them only with the negative and the cognitive phenotype. So to kind of say that all of them represent the same particular disorder would be doing injustice to the construct. And I do agree that there are multiple schizophrenias. And rather than being defensive about it, I think that is what is the essence of schizophrenia, that it is a very heterogeneous construct. It is not homogeneous in nature. And we need to embrace this heterogeneity and try to understand that because it is a disorder of disintegration, and because it is a disorder that manifests in people from various walks of life across various stages, and both at neurodevelopmental stage and neurodegenerative stage, it is going to manifest in a wide variety of clinical phenotypes. And instead of being surprised by this heterogeneity, we need to kind of embrace it and look towards more targeted interventions based on the clinical phenotype that we are seeing.
So you feel that discussing a neurobiology of schizophrenia itself is really unfair to an extent, maybe fallacious too, rather than that discussing about symptoms and their neurobiology and how they come together in a particular disorder. Is that more fair?
I do agree with you, sir, on that, because rather than going and attacking the construct which is constantly being redefined over every few decades, and I think going after the psychopathology based on the RDoC models or the high-top models would probably do more justice to our understanding of the neurobiology of this complex disorder. Yes, yes, sir.
Thank you. I hand over to Dr. Jyoti if she has any comments, and then Prajakta for taking up the questions.
Yeah. Thank you, Dr. Mohan, and thank you, Dr. Satish. It was an extremely lucid and succinct presentation. I don't have any personal questions, but I think most of the questions that I see on the chat box do not really seem to be related to
Not really
the
related
topic.
to the
So
topic.
maybe Dr. Prajakta can just take the questions if there are more questions that have come earlier.
No, sir, actually, there are no other questions. These questions are not really relevant to the topic, but then we can always get back to them later. Thank you so much,
Thank
sir.
you.
Thank you so much, sir. Thank you for such an informative lecture. Thank you to our chair people, Dr. Ashwin Mohan and Jyoti Shetty, ma'am, for removing their valuable time. So let's move on to the next session.
Thank you so much.
Thank you.
Thank you.
So our next session is on neuromodulation techniques by Dr. Shyam Sundar Arumugan. I hope I pronounced it right. Our chairpersons, I would first like to introduce Dr. Smita Deshpande. Of course, ma'am doesn't need any introduction. She is a professor of psychiatry at St. George's National Academy. Ma'am has been the professor and head at Dr. RML Hospital. She has been the research head of multiple research projects. She has developed the ISA, which we all use for the Indian scale of assessment of autism recognized by the Government of India. She has been a co-author in the tobacco cessation guidelines for the Government of India. She was also a member for developing the ICMR human research guidelines for vulnerable groups in 2017. Ma'am is on several international journal boards. She is the co-chair of the World Psychiatric Association section on genetics, and ma'am is also the international WHO trainer for tobacco cessation, and she's given lectures in multiple countries. So welcome, ma'am. I think ma'am's video is stalled, but I would like to also introduce Dr. Roop Sidana. Sir is the clinical director of Tek Chand Sidana Memorial Psychiatric Hospital and De-Addiction Center and the Prerna De-Addiction and Rehabilitation Center in Sri Ganganagar, Rajasthan. Sir has done his MD Psychiatry from the prestigious PGI Chandigarh. Sir is the advisor and the task force for COVID-19 pandemic and mental health. Sir is also the president of the Indian Association of Private Psychiatry, Rajasthan. Sir has been the past president of the IPS Rajasthan State, the IPS North Zone. Sir is on the National Advisory Board of the IAPP, and Sir has also received numerous awards like Dr. Gehlot Award, Kota Award, and Dr. Shiv Gautam Oration Award. Sir has presented, we've all heard his lectures in various national and international conferences, and his special interest is in substance abuse, RTMS, and psychosexual problems. Welcome, sir. Good evening. If one of the chairpersons can please introduce the speaker for today.
I'd request Dr. Sidana to do so, please.
Thank you, madam. Can I have this slide
Yes,
for
sir.
Dr. Shyam Sundar introduction?
The slide, sir. Is it visible?
Yeah. It's first of all, I thank the IAPP organizers to give me the opportunity to chair the session with Dr. Smita Deshpande. And our speaker is learned professor of psychiatry from NIMHANS, Bangalore, Dr. Shyam Sundar Arumugam. and he is the consultant of OCD Clinic and is an intermediate fellow of the DBT Wellcome Trust. And he did his MD from AIMS New Delhi, and he has been a clinical fellow in mood disorders, UBC, Canada. His interests areas are OCD and neuromodulation, and he has a lot of publications, more than 75 in peer-reviewed journals, and he's going to throw light on latest techniques and newer venues in neuromodulation. I welcome you, Dr. Shamsundar Arumugam, and request Dr. Smita Deshpande to introduce the topic and start the session.
Thank you, Dr. Sidana. Indeed, it is a pleasure to have such a learned speaker with us, especially on these issues of neuromodulation, which is such a recent development. Just about 8 to 10 years ago, we would not have thought of doing something to the brain just in a living and in a comfortable and in a conscious person. We were stuck with ECT and we did not know how to go further ahead from there. But now we have, and increasingly have a number of neuromodulatory techniques which do not need invasiveness. The only disadvantage that I can think of for these situations is that they need to be repeated again and again. I'm sure Sham Sundar will tell us more about it. I know that there is a disturbance going on, so I will keep quickly finish my introduction and ask the speaker to please begin his speech.
Thank you, madam. Thank you, sir. Thank you for the kind introduction. So,
yes, I'll stop screen sharing and you can share your screen.
sorry, I think there is some issue. Just a minute. I'm not able to see it. Excuse me, there's some small issue with my screen.
No problem, sir.
Yeah, now I think I'm able to share it.
Visible, sir.
Yeah. Okay. Good evening, everyone. And I thank the organizers for inviting me for this talk. So my talk will be like, it'll be on advances in neuromodulation techniques. That's what we decided to talk about. But I had a discussion with Dr. Avinash, what should I focus on? Because there are multiple neuromodulation techniques, right from like what Dr. Smita Deshpande said, right from ECT, has been there for many decades. And then we have RTMS, Tdcs, and various other neuromodulation techniques, including DBS. So Dr. Avinash suggested that let us focus on the two sort of recent but non-invasive techniques, that is RTMS and Tdcs. So I'll try to focus most of my talk on that. And if there are other questions related to other neuromodulation techniques, I'll be happy to discuss also. So yeah. So when we talk about, just like Dr. Smita Deshpande said, when we talk about intervention in psychiatry, we initially used to talk about pharmacological interventions and psychological interventions. Pharmacological interventions, as we know, focus on correcting, like it is hypothesized to correct the neurochemical imbalance in the brain. Psychological interventions target psychological aspects such as cognition, cognitive distortions, or the way we process emotions, the way we cope with situations. But recently, neuromodulation or neurostimulation, as we call it, has been added to this armamentary, where we focus on correcting the aberrant neural circuitry of the brain. So earlier, we used to talk about whether a particular part of the brain is problematic or not, like what is the schizophrenia part of the brain, or which is the OCD part of the brain, or which is the depression part of the brain. Now, we know that it is not one particular part of the brain, it's rather a particular circuit that is dysfunctional in particular disorders. So These circuits connect various various brain parts, and some of these aberrant activity in some of these circuits could lead to various symptoms. So neuromodulation or neurostimulation treatment tend to correct or aim to correct these aspects. But as we all know, these are quite interrelated because these circuits are connected through neurochemicals or neurotransmitters, and these circuits actually control various psychological aspects. So treating in one aspect may actually help us in modulating different aspects of the pathophysiology also. I'll start for, like, just to give an example, I'm talking about OCD because I'm more familiar with this particular topic. When we talk about OCD, we generally talk about the corticostriato-thalamocortical circuits. These are sort of loop-like circular pathways that start from a particular cortical region and go to a particular striatal region, that is basal ganglial region, then to a particular region in the thalamus and goes back to the same cortex. So earlier, people used to talk about one particular CSTC circuit that was involved in OCD, but now we know that there are multiple CSTC circuits, and in different patients, different parts of the circuits might be more dysfunctional. So what neuromodulation aims is to correct the activity in these particular circuitry. For example, the sensory motor circuitry, which starts from the supplementary motor area, or the dorsal cognitive circuit starts from the dorsolateral prefrontal cortex. So the neuromodulatory interventions try to modulate the activity in this particular region so that this particular circuitry activity can be modulated. So how does this neuromodulation work? So they work on the principle of neural plasticity. As we all know, the connections within the brain undergo constant changes throughout the day or throughout our lifetime. So they are not static, they always change based on external or internal circumstances. So any new learning, any new memory, or anything that happens, anything that we forget or anything we learn new is based on this phenomena called as neuroplasticity, especially the synaptic plasticity, where there is an increase or decrease in strength of connection between two neurons at the synapse is considered to be an important thing in any new learning. So this happens through the process of long-term potentiation or the increase in synaptic strength between two neurons, or long-term depression, which is decrease in synaptic connection between two neurons. So this is modulated through glutamatergic mechanisms. And neural or neuromodulated treatments are aimed to increase or have work through synaptic plasticity, where they either cause long-term potentiation or increase activity in a particular synapse, or long-term depression or decrease activity in a particular synapse. So with this background, I will discuss about the individual neuromodulation techniques. So this is a broad classification of different neuromodulation techniques. So we can start from the non-invasive ones. The most popular among them is repetitive transcranial magnetic stimulation and repetitive or transcranial direct current stimulation. But there are a host of other new interventions also possible. These are all non-invasive in the sense that you can place the device on the head above the scalp without being invasive. The second one is seizurogenic, which has been there for quite many years, which is ECT. We know that there are disadvantages of ECT. ECT, the advantage is that it has been well tested, and we know it's highly effective, especially in people with depression and also in people with schizophrenia and mania, as many Indian studies show. But the problem with ECT is that we give a current to and it stimulates to a large part of the brain, and we give a high enough current to cause seizures. So it's relatively non-focal stimulation compared to the non-invasive neuromodulation techniques, which are relatively focal and try to modulate a particular part of the brain. So because of that, the advantages is that it might lead to widespread electrical or neural plasticity, but it can cause some side effects too. The third is invasive neuromodulation, where we stimulate deeper regions in the brain, especially deep brain stimulation, which has of late found to be helpful in many patients with treatment refractory OCD and depression. So the regions that are not accessible to superficial stimulation like transcranial stimulation, we might aim to stimulate through deep brain stimulation and other newer techniques. As I said, I will focus on the non-invasive part of it. So I'll start with RTMS. So how did RTMS start? So RTMS is not a very old treatment. It started sometime around the 1985 That was the first time Barker and colleagues from Sheffield. They initially, the RTMS originated for physiological studies. Initially, people wanted to study how, wanted to stimulate animal brain. They used electrical stimulation. But the issue was that electrical stimulation was a bit difficult to penetrate the skull. So they, Barker and colleagues, formulated a device which was able to stimulate through magnetic stimulation. The advantage of magnetic stimulation is that it doesn't dissipate at the scalp or skull, and it can easily pass through the skull and go to the brain. So they said that this can be used to study physiology of the brain. But later, within a few years, that is in 1993 and 1995, people started testing the clinical implications of this stimulation technique, and RTMS as a treatment for mood disorder was being studied. So yeah, RTMS has been studied in mood disorders for quite some time, for more than 27 years. We have some studies. So that is where most of the evidence lies, but there are also studies on other disorders as well. So how does RTMS work? RTMS works on the principle of electromagnetic induction. Basically, what happens here is that this is a coil that is held over the brain or the scalp. And actually in the coil, an electrical current passes through. And when the by the principle of electromagnetic induction, when there is rapidly changing electric current, a magnetic field is produced that is perpendicular to the electric field. So here it is, electric field is produced in this direction and the magnetic field is produced in this direction. And when another conductive material is present nearby, an electrical current in the opposite direction to the original current will be produced in this conductive tissue. So because of this magnetic field, so the current goes in the counterclockwise direction in the actual coil, but in the brain tissue, which is the conductive material, current is produced in this direction. So this stimulates the particular part of the brain, and that leads to stimulation. So as I said, the electrical stimulation doesn't directly stimulate the brain. It is the magnetic field that is produced that stimulates the brain. One important aspect of RTMS is that it is an action potential inducing stimulation, in the sense that when you stimulate it, an action potential is produced in the neuron. I'll be coming to that when we discuss about Tdcs in more detail. So yeah, the advantage, as I said earlier, it is easily penetrable. The magnetic field passes through the skull easily compared to electrical stimulation, which is sometimes shunted through the scalp and the hair. Yeah, and it can be more focal. So how does it work? A single pulse produces an action potential in the underlying neuron, but if you give repeated stimulation, it produces lasting effects beyond the period of stimulation. So it changes the cortical excitability of the underlying cortex. It has been studied extensively in the motor cortex, but it has also had some effects on the other parts of the brain also. Like for example, if you give repeated magnetic stimulation at a frequency of 1 or less, that is 1 per second, or 1 in 2 seconds, or 1 in 3 seconds, it is called low frequency RTMS, where it decreases the cortical excitability in the underlying region. But if you give it at a high frequency, which is 5 hertz or 5 per second or above, it increases the cortical excitability in the particular brain region. So based on the hypothesized dysfunction in a particular brain region, it can either increase or decrease the excitability of a particular region or synapse or circuit which you want to target. So I'll just go through the process, how RTMS is being done. That might help us to understand how it's being done. So yeah, the first important thing is screening for contraindications. Our RTMS, as I said, it's a magnetic stimulation. So any metallic hardware that is in close vicinity to the coil is a complete contraindication, especially something that is less than 10 centimeters from the coil, like something like a cochlear implant or DBS pulse generator or many medication pumps, aneurysm clips. Any metallic implant in the brain is a contraindication because it can heat up that. It can also it can lead to some movement of these devices. Like MRI, this is also magnetic stimulation, so it can lead to, so these are contraindications. The other contraindication is RTMS can rarely cause seizures. So any person with high risk of seizures, especially people with epilepsy and other seizure inducing thing, it's not an absolute contraindication, but we had to be a bit careful before giving RTMS. There are some protocols which are relatively safer, which can be provided for those people. And metallic implants in other parts of the brain, like little further away, there are relative contraindications, but we had to be a bit careful while doing it. There are many RTMS screening questionnaires which are available, which help us to decide whether a person is suitable for RTMS or not. So overall, the most important thing is metallic hardware in the brain, and relative contraindication is seizure. So next is, how do we choose the target of stimulation? RTMS is generally a very focal stimulation. The most commonly used, there are different RTMS coils available. I'll be coming to that a little later, but the most commonly used coil is called the figure 8 coil. It's a very focused stimulation because it stimulates around 2 centimeter depth and around 3 centimeter squared area. So we had to be sure which area we want to stimulate. Based on theoretical rationale and evidence, we try to target a particular region which is hypothesized to be involved in the brain. But the disadvantage is that, as I said, it can reach up to a depth of 2 to 3 centimeters. So not many regions are accessible for RTMS. So for example, things like more deeper regions like amygdala, hippocampus, which are of quite of interest to psychiatry, might not be accessible for RTMS. So we tend to stimulate the regions that are connected to these regions. For example, the frontal cortex has connections to these regions, and we tend to we aim to stimulate those regions. Then how do we localize a particular target? So how do we know a particular target, particular region of the brain is being stimulated? So yeah, as I said, the RTMS is very focal, so we had to be reasonably sure which area we are targeting. So there are two basic methods. One is based on anatomical landmarks. There are different ways. One is the 1020 EEG system. Based on 1020 EEG system, for example, we can say this is standard EEG montage for placement of EEG electrodes. Based on that, we can say that like the F3 electrode position is something that is closer to dorsolateral prefrontal cortex, or the FP1 is closer to orbitofrontal cortex, or somewhere between the T3 and P3 region is closer to the temporoparietal junction. So something similar to that, we have some anatomical landmarks on the scalp based on 1020 EEG, and we tend to target those regions. The advantage is that it can be done without any additional apparatus, but the disadvantage is less accurate. But the more accurate method is neuronavigation, where we use MRI guided RTMS. So we take the MRI of the person based on structural or functional imaging. We feed that MRI into that particular device, and with that help of the device, we'll be able to accurately position the electrodes. The coil for stimulation. Yeah, but it's we need a separate apparatus, and it's generally quite expensive. The apparatus itself may cost around 30 to 40 lakhs or something. So it's that is the flip side of neuronavigation. So as I said, there are many different types of coils available for RTMS. Each has its own advantage and disadvantage. Generally, it is seen that the more deeper the coil goes, the less focal is it. And so if we aim for focality, or the more focal you want the stimulation, the figure 8 coil is the standard coil that has to be used. Yeah. I'll just show a small video. I'm not sure whether you'll be able to hear the audio. This is the figure of 8 coil, this is the figure 8 coil, which I was talking about, which nothing but two coils, two circular coils that are connected in an 8-shaped manner, and the actual stimulation happens in the center of this particular coil. So you have to place the target just beneath this particular center, and then you give a stimulation. So that's how figure 8 coil stimulation is produced. This is the most commonly used coil for clinical purposes, but as I said, the disadvantage is that it is very focal, but it doesn't go very deep. It goes up to a depth of only 2 centimeters. There are recent advances where we have deep coils available, where there are two deep coils, double cone coil, which was a slightly older one, where it's something similar to the figure 8 coil, but it is slightly angulated. As you can see, compared to the previous one, it is the figure 8 coil was actually at 180 degrees to each other. This is something like they are slightly angulated, and this can go to deeper regions. But the more recent advanced version is what is known as the H coil. This is produced by an Israeli company, and it's a patented technology. And through an array of coils, they say that they are able to reach deeper regions. So yeah, they can go up to a depth of 5 centimeters or something. And there are many studies using these coils, especially in depression and OCD, which have shown positive results. So yeah, but the disadvantage of this particular helmet or deep coil, H coil, is that for each particular region, you have to have a different coil. Like for example, there is a particular coil for dorsolateral prefrontal cortex. There's a particular coil for anterior cingulate cortex. So the same coil cannot be used for different regions. So like for depression, what is known as the H1 coil is used, which targets the dorsolateral prefrontal cortex. For OCD, the H7 coil or the anterior cingulate cortex is the target. That needs a different coil. So that's how it works. So yeah, that's about the coil. The next is about stimulation parameters. So there are different stimulation parameters which can be sort of manipulated while doing RTMS. One is the stimulation strength, like how much magnitude of magnetic stimulation you want to give. So how do we test it, whether that is adequate or not? What is known for that, we use what is known as the motor threshold. That is, because motor cortex stimulation we can easily observe. I'll be showing in a subsequent slides. Like if you stimulate the hand motor area, you can observe the hand movement. So you will know the threshold at which hand movement is observed. So that is called as the motor threshold. So you have to, if you go to the minimum strength at which at least 5 out of 10 stimuli produce specified movement in the thumb area or the hand area, that is considered the motor threshold. So that is the minimum threshold. So we sort of hypothesize that the threshold will be similar across other regions also. So when we stimulate the other region, we use the similar threshold or slightly higher than that for stimulation. So that is one thing we want to know. The second thing is what is known as frequency. As I discussed earlier, low frequency is inhibitory and high frequency is excitatory. So yeah, and the other important thing is that RTMS cannot be given continuously. As I said, when it is given continuously, it can lead to seizures, especially with high frequency RTMS, because higher the frequency, higher the chances of seizures. So we give it in something like trains, where we give it for some time, we give up some pause for some time, and then again we continue. So that is something that has to be decided. That there are some standard guidelines available based on which we can decide that. I need not go into those details. There are standard protocols available for such stimulation. So with this background, I'll be discussing about the evidence for RTMS in psychiatry. What are the major disorders? Like, as I said, most of the evidence is in depression. I think at least 60 to 70 randomized control trials will be available in depression. In, actually not in, that may be an exaggeration, maybe at least 40 to 50 would be available. So, but yeah, after that schizophrenia, then OCD, then also other disorders are also there. So I'll be focusing slightly on these 3 disorders, which has more evidence. Depression, yeah, what is it rational for RTMS in depression? Around 2000, there was a series of studies by Helen Mayberg and other people who found that there is some sort of asymmetry in activity in depression. So in the left dorsolateral prefrontal cortex, they found that activity was, it was hyperactive. On the right side, it was hyperactive. So people suggested that why not use RTMS for increasing the activity on the left dorsolateral prefrontal cortex. So the most commonly used protocol is this high frequency RTMS over left dorsolateral prefrontal cortex, so that we can increase the activity in that particular region. So later people also investigated whether we can use the other way, where the right side we can inhibit through low frequency stimulation. So that is another commonly used protocol. People later also started using bilateral stimulation, where you give high frequency on the left side as well as low frequency on the right side, so that the effect can be stronger. So that is another, these are the 3 main protocols that is commonly used in RTMS. So yeah. Yeah, so how is the coil placed? I'll just show some videos to show the standard way to place the coil is what is known as the 5 centimeter rule or 5.5 centimeter rule, where you find the area of stimulation for the for stimulating the hand motor area, and then you 5 centimeter anterior to it is the region where you have to stimulate for depression. For example, here we are trying to find the hand motor area. So I'll just pass through the video. So hand motor area, approximately, it's something like you find the vertex, which is the highest point in the skull, and go a little bit 4 to 5 centimeter lateral to it, you will find this thing. Like when we stimulate it, you can see that the hand movements are happening. So this is the hand movement area, hand motor area. So the area of the brain scalp, which has the highest movement, which is more sensitive to movement, is the hand motor area. And from that, you go 5 centimeter anterior to it. Like for example, this is the hand motor area. You go 5 centimeter anterior to it, you will find the localization of the DLPFC. That is how RTMS is given for depression. Okay, so what is the evidence? As I said, there are multiple studies, multiple systematic reviews and meta-analysis. This is just a CANMAT guideline which says that RTMS has level 1 evidence. As I said, that it is high frequency RTMS especially has been found to be helpful. And one interesting aspect of RTMS is that people who don't respond to one particular protocol might respond to the other protocol. So there are, based on that, you can try which protocol you want to give. Which protocol to give, as I said, high frequency is the most well-studied one. So that is commonly used, and it's the FDA approved protocol. The low frequency one, the advantage is that it has lower propensity for seizures, and some recent studies say that it might be equally effective. So that is another protocol that can be helpful. So what are the adverse effects of RTMS? The most common adverse effect is headache, mild local discomfort, which can be seen because when it is stimulating, it's not only stimulating the brain, it's also stimulating the scalp muscle. So there may be some stimulation and that can lead to some discomfort and pain, but that is usually self-limiting. People get used to it, and sometimes it might need some paracetamol or something, but it's generally self-limiting. But the most serious adverse event is seizure induction. Recent studies show that it is not as high to be so scared of. It's in the range of 0.01 to 0.1%. And some people say that, like one recent guideline said that it's almost equivalent to that of antidepressant medication, but still it is a possibility. So you have to be a bit careful, especially people with brain lesions and high frequency RTMS. The higher the frequency, more the chances of seizure. So we had to be a bit careful about. Contraindications we have already discussed. So with this, I'll be just discussing about, some recent advances in RTMS, what is known as Theta Burst stimulation. So this is like generally when we give RTMS, as I said, there will be a single pulse that will be provided. But in Theta Burst stimulation, instead of a single stimulation, each time we stimulate, it happens in bursts. That is, 3 stimuli are given at very high frequency together. Even the normal neuronal communication happens in some sort of bursts. So this burst sort of stimulation has been used in animal studies to study physiology for a long time. And these animal studies found that this burst stimulation is capable of producing long-lasting neuroplasticity. So based on that, people started using such burst stimulation. So these bursts, as I said, like the standard one is called Theta Burst, where 3 stimuli are given at a single point of time in a very high frequency, and this is called one burst, and that is given at a frequency of 5 per second. So 5 bursts are given per second. When we say 5 bursts, it means 5 into 3, 15 stimuli are given per second. So this 5 burst is in the theta range, that is 4 to 8 is the theta range. So that is why it's called Theta Burst stimulation. So overall, there are 2 types of Theta Burst stimulation. One is what is known as excitatory or intermittent Theta Burst stimulation, where instead of giving it continuously, you give stimulation for 2 seconds, give 8 seconds of interstimulus interval, intertrain interval, give a pause, then again give it for 2 seconds. So if you give it in such a intermittent way, it was found that it increases the excitability in the underlying cortical region. But rather, if you give it continuously without break, it leads to some sort of continuous stimulation. It leads to some sort of inhibition in the underlying region. The advantage of Theta Burst stimulation is that the overall protocol is very brief. Like for example, for the normal depression protocol for with high frequency, it takes around 40 minutes, 37 minutes to be precise. But Theta Burst can be completed within 3 minutes. So that is one advantage. The second advantage is that theoretically it has been shown to have longer lasting changes in the brain compared to high frequency stimulation. So slow off late, there has been a lot of interest in theta burst stimulation. Yeah, I think I'll not go through these videos because I think the audio might not be audible to you. So I'll just yeah. So this is one study which compared in depression theta burst to the high frequency stimulation, and yeah, they found it was a fairly large study, around 400 patients, one of the largest studies in RTMS, and they found that both are equally effective, or rather theta burst was not inferior to high frequency stimulation. So it can be used, and the tolerability was also similar. I'll skip this slide too. Yeah, that is one recent advance. The other recent advance that has happened is, as I said, deep TMS, where this particular coil is able to reach deeper regions of the brain. And yeah, this is for depression, we target the dorsolateral prefrontal cortex with deep TMS. And this is a study which compared deep TMS with sham TMS, which found that deep TMS over time, it is definitely superior to sham stimulation in decreasing Hamilton depression scores. So yeah, and there is only one study that actually compared deep TMS to regular TMS. Actually, the study is not like it compared 3 groups: deep TMS, regular TMS, and the control intervention, which received pharmacotherapy alone. Both groups were definitely superior to the pharmacotherapy group, but overall, if you see, there was not much difference between these 2 groups. But in certain outcome measures, like the response rate, deep TMS had some advantage, but in other outcome measures like overall decrease in scores, both were almost similar. Yeah, so deep TMS has some advantage, but whether it is superior to regular TMS, it's not yet proven. So now I'll come to the next disorder, which is schizophrenia. And Dr. Suhas presented a detailed description of schizophrenia. And yeah, we had a discussion during the discussion thing also whether schizophrenia is not in unitary disorders. Similar to that, we know that schizophrenia has a multitude of symptoms where different dimensions of symptoms are possible. So in the single focal stimulation might not be able to treat all the problems. So there are 2 main targets that are commonly targeted with RTMS, with auditory hallucinations and negative symptoms. So as Dr. Suhas had mentioned, the temporoparietal junction is found to be hyperactive in patients with auditory hallucinations. So we give deep RTMS over this temporoparietal junction. We give low frequency to inhibit that particular region. And there are some studies which have shown that it is helpful. The results are not as consistent as in depression, but at least there are positive results. So it can be tried in resistant patients. So this is one modality like treatment where the indication is that it's generally indicated in people for who have residual auditory hallucinations after most of the other symptoms have improved. There are studies on negative symptoms also. This is a meta-analysis which shows that RTMS, especially high frequency RTMS targeting the dorsolateral prefrontal cortex, where you stimulate the dorsolateral prefrontal cortex, they have found that it helps in negative symptoms, but we don't know how long the effects will last, because we know that negative symptoms tend to be quite persistent and chronic. So the clinical utility of this thing is still not proven. Now, yeah, so with that, I'll come to the next disorder, which is OCD, which is my area of interest. So in OCD, these are the common areas in which RTMS have been provided, the DLPFC, the pre-supplementary motor area, orbitofrontal cortex, and also medial prefrontal cortex. Earlier studies in all these areas were positive, but over time, there have been many inconsistent studies also. So like this is one of our studies which showed a negative result. Supplementary motor area, basically earlier studies found that supplementary motor area is hyperactive in OCD. So people tried to give low frequency. Early studies were positive, and many recent studies, including our study, was negative. So after that, people started looking at regions beyond other superficial regions. That's when deep TMS came. And this is one study which is relatively large study, sample size of around 100, where they gave deep TMS to the anterior cingulate cortex or the dorsal-medial prefrontal cortex, and they found that this particular treatment has been found to be helpful in improving OCD as compared to sham stimulation. So based on this study and another study, this is an FDA approved protocol for depression, but the disadvantage is that it requires the deep coil. So yeah, I think this is a meta-analysis, network meta-analysis, which we conducted recently, where we compared different RTMS protocols for say OCD, and generally what we found is the most the chances, like the most effective of these were the protocols targeting the anterior cingulate cortex or the dorsolateral prefrontal cortices. But still, as I said, the studies on other regions have not been very consistent. So if at all, the dorsolateral and dorsomedial prefrontal cortex are some regions which can be targeted with RTMS. As I said, dorsomedial means you have to use deep TMS, but for dorsolateral, we can use the superficial coil also. So yeah, these are some of the other studies that have been done. I'll just go to summarize. This is a recent evidence-based guideline for RTMS in various psychiatric disorders. What does that guideline say? Like the level A definitive efficacy is there only for depression, which is high frequency, and level B probable efficacy is there for other protocols of depression. But for disorders like schizophrenia, OCD, substance use disorders, it is possible efficacy because the studies have not been entirely consistent. So this is the overall summary of what we can say. Depression, there seems to be a role for RTMS, but in other disorders, it's still under study. Yeah, with this, I'll go to the next stimulation paradigm, which is transcranial direct current stimulation. Yeah, history of TDCS, I'll go through it briefly. Electrical stimulation has been trained in the brain for a long time. Like this is something interesting where people in Greece used to use fish that can induce electrical stimulation for treating various things like arthritis and various other things also. But actual TDCS was sort of stimulation was introduced in Italy in around 1800s by this Giovanni Aldini. He is the nephew of Galvani, who invented the direct DC battery. So when Galvani invented the DC battery, Aldini used this DC battery to give stimulation to a person with melancholia, and he said that the treatment has been effective. And after that, more than clinical stimulation, it has been used widely for in many animal studies and to study physiology. And somehow in these studies decreased in frequency, or the interest decreased till up to 2000. And it was in the 2000 by a group of studies by Prairie and Nietzsche, Prairie from Italy and Nietzsche from Germany. They showed that TDCS can cause long-lasting plasticity. It's not only during the stimulation, even after stimulation, this can cause long-lasting plasticity and long-lasting changes in the brain. So after that, people started using it. So TDCS is a relatively younger intervention, which has been there with us only for around 20 years. So overall, how does TDCS work? It's a very, very simple device compared to RTMS. It's just a simple battery that is connected to an anode and a cathode. So yeah, so this is the battery, this is the anode, and this is the cathode, and the current continuously flows from the cathode to the anode. Through the brain, it passes through. Generally, a current of 1 to 2 milliamperes is given. So it has been shown that the region under the anode, has an excitatory effect, or the excitability of the neurons in this region increases, and the region under the cathode, the excitability of the neurons decreases. So that is the advantage of TDCS, where like what Dr. Suhas was mentioning, if you find that one region is hyperactive, the other region is hypoactive, you can give TDCS with anode on one region and the cathode on the other region, and you can try to decrease or increase the excitability of that particular region. Another important aspect of TDCS as compared to RTMS is that it is not action potential inducing. Like in RTMS, every time you give a stimulation, it induces an action potential. It can be seen like when we were measuring the motor threshold, you could see the hand moved because the action potential was produced in that particular part of the brain. But TDCS doesn't produce an action potential. The neuron doesn't fire. Like this is the resting membrane threshold. This is how, this is where the membrane's potential is usually is, electrical potential. But when it reaches beyond a particular point, that is the threshold of action potential, it produces an action potential, and the neuron, the action potential, there's a spike. What annual TDCS does is, it just sort of causes membrane depolarization, or it increases the membrane potential to a slightly higher level. So the action potential can be induced very easily in this particular region. But in cathodic TDCS, just the opposite happens, where the resting membrane potential goes a bit lower. There is hyperpolarization, which so that it's difficult to produce an action potential in this particular region. So this is how TDCS works. It changes the membrane potential so that the innate action potential is either potential, the propensity to produce an action potential either increases or decreases. Yeah, so what are the contraindications for TDCS? As I said, like in generally it's a safe procedure. Metallic implants are considered to be a contraindication mainly because when it passes through the metal, it can easily shunt the current to some other region rather than the intended region. So it's a relative contraindications. And if there's a cranial hole, more current might be produced in that particular region. So that is a contraindication. And yeah, the scalp, if there is a scalp or skin lesion, you have to be a bit careful because already it can produce some burning sensation and sort of thing. So it can be, you have to be giving carefully. Rarely, epilepsy and migraine are relative contraindications, although chances of seizures are very less with TDCS. These are very rare and relative contraindication. As I said, like similar to RTM EEG system, we place the electrode based on, like for example, the same way F3 is the place where left dorsolateral prefrontal cortex is, and we put the stimulation in that particular region for TDCS also, or neuron navigation based on MRI can be used. As I said, generally our TDCS is given at 1 to 2 milliamperes and for 1 to 2 minutes. Nowadays, most people give at least 2 sessions per day, because it has been seen that when we give more sessions per day, it not only prolongs the effect of TDCS, it also enhances the effect of TDCS. At least 5 to 10 sessions, such sessions are generally given for TDCS. I'll briefly talk about the different protocols for various disorders. As I said, depression, we know that left DLPFC is hypoactive. So we give annual TDCS on left DLPFC. The cathode is sometimes placed on the right DLPFC or F4, which is known to be hyperactive, so that both can be given simultaneously. Or sometimes it's given in other regions like frontoparietal region or orbitofrontal cortex also. So this is the standard protocol for depression, where you give anodal TDCS or stimulate TDCS over left DLPFC. This slide has been presented by Dr. Suhas. So I'll, the same thing like what he said, in schizophrenia, it has been seen that the temporal parietal junction is hyperactive, and that causes hallucination, and the left DLPFC is hypoactive, that causes negative symptoms. So you can give anodal over dorsolateral prefrontal cortex and cathodal over anodal over dorsal prefrontal cortex and cathodal over temporoparietal junction, so that schizophrenia can be treated. So this is a common protocol that is used for schizophrenia, and this is one of the studies conducted in IMANS, which has found that this particular protocol helps in decreasing auditory hallucinations. There are many other studies also. Similarly, in OCD, there have been studies which have shown that annual stimulation of supplementary motor area. This is one of the studies conducted in our center, which found that TDCS has been found to be helpful. So what is the evidence of TDCS overall? Yeah, as I said that the best evidence is for anodal TDCS over dorsolateral prefrontal cortex. Yeah, that has level A evidence. But for schizophrenia and alcohol dependence, there is level B. OCD, it's still level C. So even in TDCS, the best evidence is there for depression rather than other disorders. So as I said, it is usually well tolerated. Sometimes local burns, reddening can be seen. Other side effects are quite rare. So the main advantage of TDCS is that it's less expensive. It's quite portable. It's so portable that recent studies have focused on home-based TDCS also, where people can administer TDCS by themselves. So I'll just go to two recent advances. One is what is known as high definition TDCS. The general TDCS electrode is quite large. It stimulates a large brain region. So instead of that, there is something called as high definition TDCS, where it is able to stimulate a particular brain region. So both anodal and cathodal stimulation is possible, through which you can give a highly focal stimulation. This is recently being studied, and some recent studies are there for disorders like schizophrenia and OCD, which are shown to be helpful. So this is one advance. And other advance, yeah, it's not TDCS, but transcranial alternating current stimulation. This is another new treatment. The device is very similar to TDCS. There's an anode and the cathode. In TDCS, there's a current flow directly from the cathode to anode continuously. But in TACS, the anode and cathode interchange, in the sense that it's a sinusoidal wave, like for once, the current will go from this electrode to this electrode, and then the next cycle will come from this electrode to this. It will be rapidly changing. Excuse me. Direction. The current is given in rapidly changing directions in the form of a wave. This is called as TACS, or transcranial alternating current stimulation. So what is the interesting aspect of it is that like generally in the brain, when we discuss about neural circuits, they are talking to each other in the form of oscillations. Like even when they are not, generally there's a continuous activity in the form of oscillations that is going through the brain, which happens at different frequency. It can happen at theta frequency, which is between 4 to 8, alpha frequency between 8 to 12, or gamma frequency, which is above 30. So if there are some evidence recently which has shown that the communication in particular circuits in a particular frequency is sort of disrupted in particular psychiatric disorders. Like for example, you can say that in schizophrenia, the alpha frequency rhythm has been disrupted, which can lead to something like hallucination. So what we can do is we can give TACS to sort of normalize the rhythm. So when you give TACS at a particular rhythm, it changes the brain's oscillation to the frequency of TACS tags. So based on that, so this is an entirely new protocol which doesn't stimulate or inhibit, but it changes the baseline frequency of oscillations in a particular brain region. This is a very new treatment, and it's still being studied. There are just a few studies available. So I think with this, I'll stop. To summarize, RTMS and TDCS are focal and well-tolerated neuromodulation techniques. They are capable of stimulating the superficial cortical regions. Early studies show encouraging results, especially for depression, as well as some evidence for auditory hallucinations, OCD, and substance use disorders, but their long-term effects needs to be evaluated. As I, this is the sort of difference between the two. RTMS, it's a depolarizing stimulation in the sense that it produces action potential. It can be quite focal. The one recent advance is the deep TMS is capable of deeper stimulation, and it's well tolerated, but seizures are a rare adverse effect. TDCS, it's non-depolarizing. It might be less focal, but with the advent of HTTDs, it has become more focal. It is less expensive and it is well tolerated. Okay, with this, I'll stop. Thank you. And if there is any question related to this or any other techniques, we can discuss. Thank you.
Thank you for an excellent presentation, Shyam.
Thank you.
Really extremely interesting. Just to get the discussion going, I wanted to ask you or make an observation that is it possible that there would come a time when there would be, we would hone up these stimulatory and damping down mechanisms in such a way that, suppose I am depressed, then all I have to do at home is to kind of just stick one anode on one side and cathode on one side, and have a TDCS dose in the morning, go off to work, and then come back and have another one in the evening, and my depression will disappear. Or maybe when I feel that, maybe hopefully a time will come when a person has auditory hallucinations and is able to control them as well. maybe through TDCS or whatever, because that would be a home-based treatment which would integrate our patients into society. And ultimately that is our aim, isn't it?
Yes.
To give our patients as good a life as possible. You have talked about these in modulatory systems, but I presume these are only for one, what shall I say, one set of treatment for one episode, whereas our illnesses are prolonged and they come and go. So can you tell us a little bit more about what has happened for people who get repeated episode? Do they get the equal amount of advantage with every episode? Or is it that the advantage waxes and wanes over time? Have any studies been done on that aspect?
Yes, thank you, ma'am. Thank you for the interesting questions, ma'am. Regarding the first question, ma'am, already it's actually people are doing it, home-based treatment actually. In many countries, it has gone to such an extent that people are using this TDCS device to sort of enhance their cognition and for various other things also, for which might be quite a little bit going into the unethical domain also. So so its safety has not been established, but TDCS is generally a safe treatment. So that is something that can be thought of in the future because it's a very simple device. So it can be thought of. The second question, ma'am, that's also really important regarding the long-term treatment, because we don't know how long this plasticity will last actually. So there are some RTMS studies which say that the effect can last up to 3 to 6 months. But I'm not very convinced, because based on my experience at least, we see that patients do relapse after 3 to 6 months. Two things have been done, ma'am. Like many people use pharmacotherapy to prevent relapse, just like in ECT. But sometimes there are maintenance protocols also. We have also used it in rare patients, but these are not established or studied protocols. Like some patients are there who comes once in a month. We give 2-3 sessions and then they do well for a month, and we continue like that for some time. So yeah, that has been tried, but their safety and as well as efficacy has not been established, and there's no standardized protocol also.
So there are no RCTs. Far from having a lot of case studies or case series, there are no RCTs.
No, ma'am, no.
But do you see a time in the future, because you are our future, do you see a time in the future when these neuromodulatory techniques would be the mainstay of treatment rather than pharmacotherapy?
I'm not sure, ma'am. Actually, to be frank, actually, I'm not very sure whether we have
a difficult question.
In the sense, because I still think that we don't understand the neurobiology of any psychiatric disorders to such an extent where we'll be able to do so, and whether it will be such a simplistic explanation where we can be able to do so. So I think, at least as of now, definitely pharmacotherapy is the mainstay. These are just add-on treatments, I would say. Maybe in the future, but there are multiple things, as you said. What about the long-term treatment, and whether it will be helpful for all types of symptoms? All these issues are there. So yes, ma'am, I think it's I'm a bit skeptical about the whole thing.
Dr. Sidhana, if you have any questions, and then I'll hand over to Prajakta.
Dr. Sidhana, if you have any questions.
So, taking on from where you left it, Cham, you say that you're feeling a little skeptical about these treatments. I'm not. Actually, I feel quite hopeful about them, because our psychology colleagues used to use, some kind of mild modulatory techniques for ages, maybe to induce sleep, maybe to do other things.
Yes.
And maybe they were just symptomatic, but they worked.
Yes.
So let's hope that this also starts working and it shows us a new direction as time goes on. Yes. I think Dr. Sidhana was saying something.
Yeah, but I think we've lost him. He's lost connection. He's not here anymore. All right. We don't have any more questions in the questions in the group.
Okay, then I have one more question then, just to keep the discussion going. Can you think of some other way in which these, the coil placement or the type of electrical stimulations that are given could be honed for specific symptoms and specific illnesses. For instance, in OCD, you know, when there are sort of runs of thoughts, or the obsessions become especially worse, could there be a pocket stimulator which could kind of help over that little acute thinking problem?
Ma'am, actually, yeah. DBS is an option for that actually. So that is some research that's going in DBS also, where like generally deep brain stimulation, where the electrodes are implanted very deep into the brain and that is connected to the battery, and through that we give the stimulation. Generally, it gives some sort of continuous stimulation. But recently there is something called as adaptive stimulation, where like when the particular activity in a particular part of the brain goes beyond a particular threshold, it will sort of stimulate only during that particular time. So if you are able to find that, like for example, the OCD thought is associated with a particular sort of frequency of activity, and when we feel that that frequency is increasing, you can make the DBS give an adaptive stimulation during only those particular times. That can happen for hallucinations or various other conditions also. So that is some research that is going on. It has been found to be helpful in Parkinson's disease, but in depression, there is some study that has been shown that it might be helpful, but that is something advanced that is happening right now.
And deep brain stimulation, you know, placing of electrode, I don't know how many have really got it done for OCD. You know, Parkinsonism is something very clearly oppressive and, you know, very clearly seen. Whereas OCD is something that can't be seen. So one can never really make out the distress of the patient.
Yes, ma'am. Actually, that's a conflict of interest because I am currently conducting a study on DBS and OCD. So, yeah.
So how many people did you ask, and how many people agreed, consented?
Yes, ma'am. That's a major issue. Not everybody, because we are trying in completely refractory people, basically. And even then, like there's a lot of doubts, because for psychiatric disorder, surgery is something, it's a major decision. They take some time to decide. Yeah, but among the psychiatric disorders, the evidence is more consistent for OCD. Yeah, depression, there has been some inconsistent result, but for OCD, there has been relatively consistent. But yes, ma'am, as you said, that it's only a small population that has been studied with DBS right now. Not many people have studied, but it has shown in treatment refractory, they have shown that around 60% of people improve, which is good for a treatment refractory population.
Yeah, but only 60%. So
that's there.
Dr. Sidhana, I'm sorry,
Yeah.
you dropped off. So if there is
Now I have a question to Dr. Shyam Sundar, and rather his views that most of the postgraduate departments in India still haven't got the RTMS as a treatment modality. Only about 25% of the departments, probably that's a very high figure, 25% of the departments, but 75% of the departments don't have a RTMS or any neuromodulation facility, and the faith in the treatment as such is very low. And many of the professors in the state-owned medical colleges still see neuromodulation techniques as a placebo therapy being used in private practice. And unless there is, even though there is a evidence level 1 for treatment of major depression refractory to first or second antidepressant failure, and the results are good, still it is not being used in the way it should be used. I have been following this for the last 10 years, and even the centers of excellence where the huge budget is there for psychiatry, using neuromodulation techniques for research and for therapy are still very few departments are doing it. So to make this neuromodulation techniques popular and acceptable by all of us professionals is a very important task which need to be done. Your comments, Dr.
Yes, sir, I can tell you that the cost is a huge issue, especially in government colleges, especially for treatments where absolute clear-cut evidence is not there. And second thing is that the number of manufacturers are very few. So the government rules are very definitive about these things. I can tell you because we went through it ourselves. But there were other questions as well. So Sham, I'll leave you to it. I won't interrupt now.
Yeah.
Can we have this as the last question because we are running slightly short of time?
But Prajakta, there were a couple of questions from the audience as well.
There is
Are
one
there any
question.
studies in child and adolescents? And there was one more.
Yes, ma'am. In children and adolescents, earlier people were a bit hesitant to use RTMS and TDCS, but there are recent studies which have shown that it is safer, but it's not something like it's not a first line treatment or something like that. It's not that well established in RCTs or sort of thing, but there is some experience of using it now. So no major issues have been found. So
please answer Dr. Siddhana's question. That
yes. Yes, sir. I think it is something like, one aspect is, yes, ma'am said that it's quite expensive, especially RTMS. Like, the machine, the device with the thing might cost around 30 to 40 lakhs. So that is one possibly bottleneck because of which it is this thing. And the other thing is, in what patient population do you want to use it? That is the another thing. Like the questions that raise in like, how long do you give the treatment? What about the long-term management? But I think there is a subset of people for whom it might be helpful. Like, for example, for acute efficacy, at least in depression, definitely that has been found to be, it's quite well proven, both RTMS and TDCS. So that is one thing. And and even for auditory hallucinations, there is some efficacy. So yeah, but for other disorders, it's still experimental, but at least these two indications are there. But the cost and I think that is one major factor. And the skepticism, I think, is coming from the what about the maintenance treatment? That I don't think we have answers to it right now.
TDCS is of the two much cheaper, so TDCS may be a more achievable target.
That definitely would be much more achievable, yeah, and it's much and it has less side effects also. Yeah, in that way, I think TDCS would be more scalable.
There was one more question from the audience.
Question which says that Dr. Majid is asking, what about the higher current TDCS, say 5 milliamp current?
Generally, like as of now, safety is proven up to 2 milliamperes. Some people use up to 4, but I'm not sure regarding that. But generally, safety as of now, it's about up to 2 milliampere is the standard.
There's one more question on how to get the equipment, but I think they'll figure it out.
Yeah, there are lots of manufacturers available, and most of the manufacturers are from European countries and also Europe. But there are some Indian manufacturers also available.
Yeah, and TDCS is very much affordable and cheap to buy.
Yes.
So if it can be used at home, then definitely it can be used in the departments. And I can see that there would be many more, people opting for TDCS than RTMS.
Yes.
Dr. Shamsundar, in my early part of practice, 80s and early 90s, there was a manufacturer in Bangalore. They used to supply brain polarizer and sleep therapy machine, which were akin to TDCS, but different names. And they claimed that one of them is activating for negative symptoms of schizophrenia, and the one of them is used for sleep therapy. And I used it for initial part in my practice, but I didn't find good results, and I dropped out. But do you have any idea whether this almost same?
I'm not aware, sir. I'm not aware of this particular thing, whether it is the same or not. But
to TDCS or not? Question is the TDCS, the portable machine, and the HD TDS. How different are they in efficacy? Because there is a lot of difference in the cost.
Yes, sir. Actually, to be frank, I'm work more on RTMS than TDCS. But from whatever I'm aware of, I'm not completely sure whether about the device you were talking about, whether it is similar to that or not. Second question is HT-TDCS. It's a very novel sort of new treatment that has come. Only now studies have started coming up in that. Advantage is its focality, definitely. I think because, I don't know, so like one way to look at it is like if we want to target one particular focal area, then HT-TDCS might have an advantage. But as in many psychiatric disorders, as of now, we want to stimulate a larger region. Then I think the regular TDCS should be adequate. I think for routine clinical use, as of now, the routine conventional TDCS should be adequate. HT-TDCS is currently still in the research phase, is what I believe.
Yeah. Thank you.
I'm sorry to interrupt this engaging conversation, but we're running short of time. Thank you, chairpersons, for such wonderful questions. And thank you, sir, for such an illustrative and informative session.
Thanks a lot for inviting me.
Thank you. Thank you.
Thank you very much for a very educative talk and really interesting talk. Thank you.
Thank you, everyone.
Thank you. So let's quickly move on to the next session. 1 second, let me just share screen. So our next session is on epilepsy and psychiatry, what most postgraduates should know. The speaker is going to be Dr. Nishant Coil. Let me introduce the speakers first, our chairpersons first. Our first chairperson is Dr. Shashi Rai. She's a consultant psychiatrist in Lucknow. She's the director of Sambal Drug De-Addiction and Psychiatric Center in Lucknow. She is the chairman of the Richmond Fellowship Society India, active member of various psychiatric societies. Her areas of interest are the women mental health rehabilitation and community psychiatry, child and adolescent mental health. She is a member of the Iabp and chairperson of the Women Mental Health Iabp. Welcome, ma'am. Our second chairperson is Dr. Niti Sabru. She's done her MD from Nair Hospital, Mumbai. She has more than 30 years of experience in practice. She's trained in the camps of the child and adolescent psychiatry in the NHS UK. She's a child and adolescent psychiatrist in Mumbai, sub-investigator and co-investigator of multiple multi-centric drug trials. She conducts various workshops in Mumbai on parenting, sex education of students in schools in psychiatry. She is a member of the Global Clinical Practice Network, the internet-based field studies for the ICD-11 mental and behavioral disorders. Welcome, ma'am, and thank you for stepping in last moment for being our chairperson.
My pleasure and honor. Thank you so much.
If one of the chairpersons can please introduce the speaker.
Dr. Niti, will you introduce the speaker?
Yes, definitely. I'll do that. My pleasure. Good evening. Thank you so much, Iabp and BPS, for giving me this opportunity. Dr. Nishant Goyal, as you can see, is MD, DPM. Can you hear me?
Yes, ma'am.
Okay, MD, DPM, designation professor of psychiatry, is in charge and Center for Child Adolescent Psychiatry, FMRI Center and Center for Cognitive Neurosciences. He's in charge academics. His affiliation is to Central Institute of Psychiatry, Ranchi, India. He's a program coordinator, mental health and psychosocial support during pandemic for CIP Ranchi. Program coordinator, CIP Digital Academy. Joint secretary and founder member, Neuromodulation Society, India. Review editor of Frontiers in Psychiatry, Child and Adolescent Psychiatry. Areas of interest, specialization, child and adolescent psychiatry. Non-invasive brain stimulations. Public mental health. Number of publications, 90+. Wow. Awards and fellowships, 20+. So welcome Dr. Nishant Goyal, and he will be talking on, just a minute, I think I lost the topic. He is talking on epilepsy and psychiatry, what postgraduates should know. I think it's a very interesting one. So let's hear Dr. Nishant Goyal. Thank you.
I'll just talk about the topic a bit. First of all, good evening, and I thank the organizing team for having invited me as a chairperson in this August gathering. The topic, as ma'am said, is epilepsy and psychiatry, what the postgraduate should know. As well as we are all aware that about more than 50 million people worldwide are suffering from epilepsy. And in our own country, there are 10 million people. That's about 1% of our population. So to my mind, because already we are a little late, 5 points I think is very important. What the PG should know. First and foremost, they should know how to diagnose and differentiate from the various pseudoseizures and the mimickers. The drug interactions, which are very important. Then at what age the drug should be given and which doses should be given, because 1.5 million females in the reproductive age group are suffering from epilepsy. And the most important, what I feel, is the pocket of the patient. That is also very important, because a drug might be very good, but if the patient can't afford, and psychoeducation, of course, of the patient. So this, every postgraduate student should know these points. And we have a very illustrious speaker, Dr. Nishant Goel. So I welcome you, sir, for your talk.
Thank you, ma'am. Thank you, ma'am. Can you all hear me?
Yes, sir.
Can I go ahead, Prajakta? Yeah.
Yes, sir.
So allow me to share my screen, please.
You are a co-host, sir, so you can do it.
Yeah. Just give me a moment.
Yeah.
So I guess my screen is visible to all of you.
Yes, sir.
Yeah, it's visible.
So, as today is mine is the last talk for today's session. So the topic has already been introduced. So I would first like to acknowledge and thank the chairpersons for giving a kind introduction. Acknowledging IABP, BPS, Rajesh Nagpal sir, Venu boss, and Dr. Avinash, and definitely my friend and my colleague, Dr. Sai Krishna Tikka, who helped me making this presentation. At the outset, Shashi madam, I would like to take a little bit of detour when it comes to what I'm going to cover in the next, say, 40 minutes. Is not about, what the postgraduates need to know about epilepsy or the management of epilepsy. The topic which was given to me is epilepsy and psychiatry. So what I'll try to do is, what is the interface of epilepsy and psychiatry, how they are associated with each other, and how, how epilepsy, various type of epilepsies can present as psychiatric manifestation, or what are the various psychiatric manifestations associated with epilepsy, and definitely a little bit about the management. So before dwelling into the topic per se, I have few questions which I want to pose, to all of the people who are actually listening to us today. So whether it's true or false, there are a few true or false questions. Is it normal to feel depressed when we know that we have epilepsy? Do you agree that suicide is a major cause of death in epilepsy? Do you agree that depression can be caused by anti-epileptic medication, use of anti-epileptic treatment? And can feelings of anxiety and or depression be a manifestation of seizure? So on the backdrop of these questions, I would first like to start my presentation. I would like to quote Carl Friedrich Fleming, who says that epilepsy and mental disorders are two states of illness of very closest relationship. They represent identical pathological condition in two different areas of the nervous system. So this particular phrase has a lot of understanding and meaning to it. And how and why we will see as I'm going to progress in my presentation. So in the next few minutes, we will be talking about how to discuss various psychiatric conditions associated with epilepsy, their clinical course, their response to treatment, presence of any underlying psychiatric disorder, or their temporal association with the occurrence of a seizure. We'll see how presentations of seizures can be misdiagnosed as a psychiatric disorders, and what are the various behavioral and psychiatric adverse effects of typically used anti-epileptic medications. So Shashi ma'am has already given an introduction to the extent of epilepsy. It is, as we all know, it is one of the most common neurological disorders. In India, she has already talked about how many patients are actually suffering from epilepsy. We know, and now that a lot of literature which says that psychiatric comorbidities are present in around 70% of the cases of refractory seizures. So they go hand in hand. And not only epilepsy, if it is associated with psychiatric sequelae or problems, it may lead to significant impairment in functioning and quality of life in an individual. So I'm sure most of us, we know what are the various, how do we classify seizure based on seizure types. We have focal, generalized, a combined type. Then we have various syndromes. And then there are various etiologies which are associated with epilepsy, starting from genetic, structural, metabolic, immunological, so on and so forth. And at the same time, they're associated with sweet comorbidities, which is very, very common in cases of epilepsy. Now, what I have tried over here is to understand psychiatric manifestations as per the semiology of a seizure, or in other words, we call it ictus. So whenever a seizure occurs, it actually, or it usually goes through 4 different phases. I'm sure most of us are aware of it, and we are taught in our postgraduate days. So the ictus is preceded by a condition called prodrome. I'll tell what it is. Then the ictus proper, or the seizure proper, which is followed by the post-ictal period. And then between the 2 ictus, there is the interictal period. So what we see is that this ictus, as I said, can be seen as a primary generalized seizure or a focal seizure, which may or may not lead to generalization. So we'll see gradually how it happens. Literature says that around 30 to 50% of patients with epilepsy, they suffer from some or the other psychiatric disorders in their lifetime, and it has got significant implications when it comes to diagnosis and management. And it gives a good opportunity for us to understand the organic basis of various psychiatric disorders. Epilepsy and psychiatry, as I said, that they are like a vicious cycle. Epilepsy is like a brain disorder which is associated with causation of various psychiatric disorders. Psychiatric disorders, when they are like they have various cognitive dysfunction which predisposes the individual to have or present with epilepsy. So it's a vicious cycle, as you can see from this particular figure. Coming to the history of epilepsy and psychiatry, I'm sure many of you might be knowing in ancient Rome, epilepsy was also named as morbidus lunaticus. So it was first seen as like morbid lunatics who were known to be having epilepsy. Hippocrates recognized that brain was the seat for both falling sickness and madness. Falling sickness was epilepsy, which was given a name during that particular era, and they both were related to phlegm. Fallray first gave the concept of folie epileptique. Morel, I'm sure we all know, Morel has also given various psychiatric conditions. He's talked about epileptic character. Sommer in 1880 talked about post-epileptic insanity. And finally, Emil Kraepelin in 1923 had clearly mentioned in his writing that epilepsy patient can undergo a change in personality, and they suffer from a predisposition for development of psychosis. So you can see how common it is associated, or they are associated with each other. When it comes to the description of psychosis with epilepsy, one of the first controlled studies was basically given by P. Floor Henry, which was published in Epilepsia way back in 1920s. So the article was called as Psychosis and Temporal of Epilepsy, a controlled investigation. This is the first investigation on 100 years back which was published. Now coming to the neuropsychiatric aspects of epilepsy, how epilepsy is associated with various psychiatric conditions. Depression, around 10 to 50%. Suicide, as I asked this question also, it is associated with around 1 to 2%, or 1 to 1.2% of individual. Anxiety from 15 to 30 or 40%. Epilepsy is also associated with alcohol and other drug abuse disorders, ranging from 12 to 30%. Psychosis from 3 to 9%, so on and so forth. If you see the overall comorbidity, or the the incident of comorbid epilepsy and psychosis is around 30 to 60%. And I can vouch for it because I have been working in a setup where we have a standalone epilepsy clinic which happens every week, and I'm in charge of the epilepsy clinic for the last 14 years. And I can completely agree to this, working in a WHO project also, that it is true. The comorbidity is very, very huge, and it's a huge number when we are seeing such cases. So I'll not go into the putative mechanisms of that, but they say that they both increase the vulnerability to each other. They share the same common brain substrate, and there is definite role of anti-epileptic treatment for epilepsy and at times epilepsy surgery, which may be associated with causation of various psychiatric disorders. So this basically particular figure, it talks about, or it kind of sums up the various mechanisms. You can see from here that there are some functional factors which are largely reversible. Then there are some clinical and diagnostic features. There are morphological factors. Depending upon the site of lesion, we have epilepsy, and depending upon the side of the brain where the lesion is, people will have a particular set of psychiatric disorder. For example, temporal lobe epilepsy, when it is affecting the left side of the temporal lobe or the left hemisphere, it is associated with depression. Or if it is on the right side, it is associated with elevation of mood or manic-like symptom, just to give an example. Now, as I said, classification of psychiatric aspects of epilepsy can be broadly classified. There are different classification. I'll stick to the most commonly used classification. So they talk about something which is related to ictus. As I said, it can be ictal, periictal, interictal, and then it is what is associated with the treatment of epilepsy. So there are other classifications. Recently, around 7 or 8 years back, Krishnamurthy and Trimble, they also gave another classification of neuropsychiatric aspects of epilepsy, where they basically categorized, the comorbidity as per the problem or the psychopathology which is presenting as a epileptic seizure. Suppose a person who is having anxiety as the ictus or hallucination as a complex partial seizure. So we'll come to it. We'll see how it happens. Then there is an interictal psychiatric disorders which are also specific to epilepsy. We'll talk about it as we move on. So first, coming to the pre-ictal state, which is commonly known as prodrome. So prodrome basically refers to a set of symptoms which indicate the onset of an attack. It is not an attack minded. Onset of an attack. The duration is usually variable. It can last for minutes to up to days. And the most common content of prodrome, which is reasonably stereotypic, is irritability, sad mood, cheerfulness, headache, loss of interest or appetite, anxiety, and restlessness. We have found that any epilepsy or any psychiatric conditions during the prodromal period, it is not the beginning of seizure. It might last for a few hours, commonly seen as anxiety, depression, and there will be subtle changes in the mood, behavior, and thinking. Typically, when you do EEG during this time, you will not find any ictal or interictal changes. You don't find psychosis in the pre-ictal or the prodromal phase. It is very, very unlikely that you get psychosis-like presentation in the prodromal phase. Moving on to the ictus. Ictal psychiatric manifestations are further classified into two. First is the psychic aura or the ictal psychic symptoms. And the second one, which is very, very commonly misdiagnosed as a functional psychiatric condition, is a non-convulsive status epilepticus. It's a status epilepticus, mind it, which doesn't present with a convulsion. So how it happens, we'll see. Now coming to the psychic aura, I'm sure there is a backdrop that we all know that what is aura, what is SPS, what is CPS, like that. So aura can present as a psychic manifestation. And mind it, 25% of the epileptic aura, they present with psychiatric symptoms. They are associated in the EEG with discrete discharges. The person is usually aware and responsive during such phenomena. They usually occur as a part of simple or complex partial seizure, and they may herald the onset of secondary generalization, which is quite common. Around 60% of the psychic auras, they go on to develop secondary generalization or secondary generalized tonic-clonic seizure. Now, aura, as per the phenomenology, can be divided into various types. You can see over here, dysphasic, associated with language, dysemnestic, associated with memory, cognitive, affective. It can present with perceptual abnormalities like illusions or hallucinations. Now coming to dysphagia. The commonest type of dysphagia as an ictal manifestation is a non-fluent dysphagia whenever there are abnormal discharges coming from the Broca's area. How do they present? They present with repetitive vocalization with formed word. Aya, 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 gaya, gaya, gaya. So these are the words a person will start uttering. And it may occur from the affection of non-dominant temporal lobe, that is the left temporal lobe. Then ictal dysamnesia, very commonly we used to read when we were students, they present with temporal lobe seizures, particularly from the right side. They present as dia phenomenons, dia vu, dia pense, dia intendu. So these are various dia phenomenons. I'm sure in various books of psychopathology and phenomenology, we know all what are these. So I'll not go into the details of what are the dia phenomenons. Cognitive symptoms as a seizure presents as a oneroid or a dreamy state. People have altered sense of time. They do present with depersonalization and derealization like phenomenons. And there is a concept where they say that I'm forced to think or I'm forced to act. These symptoms are commonly associated when the discharges are coming from the mediobasal or neocortical temporal lobe seizures. Affective aura usually presents with ictal fear. So fear as an aura has been recognized long back. It is even also written in the book of medicine written by Sushruta. So he has mentioned about ictal fear. It is around present in 15% of the patients who present with partial seizures. And as I said, it is one of the most common emotion when it comes to the ictus. Around 20% patients, they also have ictal anxiety, and this is when you have to actually rule out clinically as well as through investigation through non-epileptic panic attack. It is a very common, what to say, differential diagnosis, ictal anxiety and panic attacks. Then ictus can present with either depression or ecstasy. Again, there's something called unio mystica or ictal sexual symptoms, which are also seen. So what I mean to say that ictus can present in any form or any ways. A seizure can present with any psychopathological manifestation we read in various books. It can present with perceptual problems like illusions, things are seen smaller, bigger, nearer, farther. So anyways, people present with hallucinations of all modalities. Some books say that visual hallucinations are more common, but not like that. Any modality hallucinations can be present as an ictal phenomenon, can be elementary, can be complex. Most commonly, the implicated regions are temporal and parietal region. Then associated with or ictal manifestations of psychiatric symptoms, there are some newer concepts. I'll not dwell into the details of these newer concepts. They're not that new, around 10 years old. People are now talking about that complex partial seizure psychosis can be divided into three. One is the elementary sensory phenomenon, another is a complex sensory phenomenon, and there is an integratory phenomenon when the two things are integrated with each other. A few words about the non-convulsive status epilepticus. As I said, it is one of the most commonly missed situation in various psychiatric settings. I can tell about my clinical experience. I have also missed an NCSC in at least 5 to 6 cases in my entire career. So this is a prolonged seizure activity in the absence of major motor signs, as we all see in a generalized status. The definition is the same: more than 30 minutes of ongoing epileptic activity. Broadly, it is classified into 4 types. One is called the absence status, which is more like a generalized status. Then I'm sure many of you might have seen something called simple partial status or epilepsia partialis continua, where in clear consciousness, a person continues to have motor manifestation or maybe sensory manifestation. Then you have complex partial status and status epilepticus, which is happening in coma. Excuse me. So as I said, non-convulsive status can be focal or generalized. I've already talked about it, what it is all about. Focal non-convulsive status is more common than the generalized non-convulsive status. So this we have to understand and realize. Now, there are various variations when it comes to focal or generalized non-convulsive status. For example, absence status epilepticus or absence seizure presenting in status, there will be clear-cut evidence of impaired consciousness with behavioral changes like disorientation, decreased spontaneity, slow speech, rhythmic blinking, and maybe at times myoclonic jerk. And you do EEG at that time, you'll clearly get 2 to 3 hertz back and slow wave pattern. Simple partial status, as I mentioned, the consciousness will be there. In complex partial status, there will be impairment of consciousness, which is typically known as twilight state of epilepsy. When a person will behave like a psychotic person, where there will be confusion, strange behavior, people will have automatisms, so on and so forth. And you will get EEG changes when you do EEG of these individuals during the time of status epilepticus. So I'll not go into the details of complex partial status. I find it too complicated. This takes me to the next part, which is the peri-ictal psychiatric manifestations. Peri is around, ictal is around the ictus or around the seizure. So this is a period where it is just before or after the ictus and applied when there is insufficient information to know when the ictus is starting or ending. So they include some of the prodromal symptoms, some of the peri-ictal symptoms, or some post-ictal symptoms as well. Coming to the peri-ictal psychiatric manifestation, the most commonly described one is confusion. Post-ictal confusion is basically a prolonged, I'm sure many of you or maybe most of you, you must have given ECTs. So just after the ECT, you get something that a person is confused. This is a typical manifestation of post-ictal confusion, where a person might have bizarre behavior and may or may not have amnesia for that particular period. If you do EEG, you will find continuous or repetitive ictal burst during that particular time. Thus, the duration is very variable. It might range from 30 minutes to 36 hours. Consciousness, as I said, is impaired, and the person might be having amnesia for the event, and it might be associated with hallucinations or transient delusion, which a person doesn't remember once there is complete regaining of consciousness. You will not get any clear cut, but some non-specific EEG abnormality. Now, why this is important? Because post-ictal psychosis has a particular criteria to describe. The criteria, which is characterized by an episode which is manifested immediately upon a seizure or within 1 week of the return of normal mental functions, lasting for at least 24 hours with a maximum period of 3 months. Associated with clouding of consciousness, disorientation and delirium, delusion, hallucination in clear consciousness. And you have to exclude that it is not because of any anticonvulsant toxicity, interictal psychosis, any EEG evidence of status, history of head injury, or alcohol or drug intoxication. So this criteria is very, very commonly called, and I'm sure all of you must have read during your MD days, or you must be reading when you, if you are doing MD, it's called Logstein and Toons criteria. So very commonly asked during exams also, what is the Logstein and Toons criteria for post-ictal psychosis? So they have given one of the first descriptions of post-ictal psychosis. Psychosis presenting during the post-ictal phenomenon. Studies have found that it mostly involves males. Frank psychotic symptoms are there with some confusion here and there. Primarily, misidentification delusions are common. People will also have abnormal mood. It can happen both ways, mania or depression. People often attempt suicide during this phase. This is the time where suicide happens during the course of epilepsy. People can also present with stupor, catatonia. They have array of hallucinations, and you will get EEG dysfunction, which is clearly shown when you do the EEG recording at the time. Another concept associated with psychosis and epilepsy very often is something which was given by Henrik Landolt. It is called forced normalization. I'm sure the seniors and my colleagues, or maybe juniors, students who are listening to us, you will definitely get a short answer in your exams. It's very, very commonly asked. It's very simple. Periictal psychosis concomitant with decreased seizure frequency. It was the first thing which was described by Landolt, that whenever psychosis increases, seizure frequency decreases. But it has been found that it can be other way around. So classically speaking, there is a mutual antagonism between the presence of epilepsy and psychosis. It is more of an EEG phenomenon, which was described previously when they say that the EEG becomes normal or partially normal as compared to the previous one if psychosis sets during the post-ictal period. So Tellenbach has also named it as alternating psychosis. There are some recent observations or some new things which have been described in the context of forced normalization. They say that EEG may or may not necessarily be normal. It can also be seen not only in psychosis, but in depression, mania, or anxiety disorders. There is an alternating pattern. They are more commonly associated with generalized seizures as described by Landolt. It may sometimes be provoked by introduction of anti-epileptic drugs. So people have talked about it. A very avid neurologist from India, Krishnamurthy, has raised some interesting questions about its trait-like character as well. And they have given a new criteria, which is a detour from the Loxden and Toon's criteria. It's called Krishnamurthy's and Trimble's criteria. This has been published in Epilepsy and Behavior. I'll not go into the details of this. So I've already talked about, alternating psychosis, which basically they changed the term from forced normalization to alternating psychosis. So I'm not going to go into the details of this. Other post-ictal psychiatric or psychic phenomena can be depression, mania, or anxiety, which can be seen. People might also present with twilight state or a fugue-like state. It is seen but less described. So there are always a confusion. What is an epileptic fugue, whether it is a ictal phenomenon or a post-ictal phenomenon. And there is the researchers have finally found that it's an admixture of ictal and post-ictal changes. And that the term which is describing this is also known as poriomania. If you put this poriomania, you'll get some good literature on this. And as I said, there are no clear-cut systematic studies. Now, a question arises that can aggression or violence can be described as a manifestation of seizure? Studies say very rare. How to differentiate? The aggression of seizure is usually spontaneous. It is non-goal directed, as opposed to the goal directed aggression seen in psychosis. And you will get some stereotype movement if you take history. It does not involve integrate skills or purposeful behaviors. And people, they have unintentional aggression that occurs when someone attempts to restrain or assist them. It might present with repetitive limb movements, shouting, spitting, shoving, pushing, something like that. So people have talked about some neurobiology of post-ictal phenomena. They talk about neuronal exhaustion, acute neurotransmitter depletion, increase in the extracellular potassium, which is very commonly associated with psychosis-like phenomena. There are other things. I'm not going to the details of those. Then briefly about interictal psychiatric manifestation. What is happening to the psychiatric symptoms between two episodes of seizure when a person has completely become all right when it comes to epilepsy? They are commonly subdivided into two types. One is a schizophrenia-like psychotic disorder and certain type of personality disorders. Very commonly, people describe something called Gestalt-Genschwein syndrome. We'll see what it is. Now, schizophrenia like psychotic disorders, it is seen in around 7% of adult cases with epilepsy. It is very, very strongly associated with temporal lobe seizures. Three types of schizophrenia like psychosis has been described by Slater and Beard. They basically describe them as chronic psychosis with recurrent confusion. Then another one is chronic paranoia like situation. And thirdly, hebephrenia like phenomenon. After that, Brunes in 1971, they gave 4 groups. Apart from paranoia, mental regression, affective symptoms, they also talked about short confusional state presenting in the schizophrenia like psychotic disorder phenomenon. Personality disorder, there is very little research which is available on the comorbidity of personality disorder in epilepsy. But consistently, we have found high prevalence of borderline personality, histrionic personality, dependent, and at the end of the day, mixed personality. The problem with studies is they have never used a control group whenever they are looking into the prevalence of comorbidity. So it is very difficult to generalize the findings. Second, on Gestalt-Genschwein syndrome. So Gestalt-Genschwein syndrome is a situation which is associated with chronic temporal lobe epilepsy, where a person presents to us like a serious individual who lacks humor, circumstantial, hyper-religious. He's or she is over-inclusive. I'm saying he because mostly seen in males. He is over-inclusive for doing any or everything. There is a lot of viscosity when it comes to talking, writing, and there is hypergraphia. They write a little bit too much and the writing doesn't have any meaning. So again, as I said, psychosis can be seen across various phases. So there are some changes when it comes to EEG, epilepsy. So I'll not go into the details of this. Then talking about mood swings and epilepsy. Mood swings and epilepsy might present with a situation called interictal depression, which is the most common comorbid psychiatric condition associated with epilepsy, with a lifetime prevalence of up to 6 to 30% in population-based studies and up to 50% in studies which are done in centers like ours. They have a patient with epilepsy, they have a 5-fold increase in the depression, in the incidence of depression, more commonly seen in temporal lobe epilepsy, who present with intractable complex partial seizure. So they present, like a chronic depression. The duration is very long. The course can be intermittent. There will be ups and downs. A few years back, Blummer gave the concept of something which is very similar to depression. He named it as interictal dysphoria or interictal dysphoric disorder. He claimed that up to 50% of the patient with epilepsy seeking medical care, they actually suffer from interictal dysphoric disorder. So dysphoria, as we all know, is a combination of mood, anxiety, and some somatic symptoms or irritability. So we get the dysphoria symptoms. So there are various types of symptoms. As I said, affective symptoms are there. Then suicide. Suicide has received very little attention to us psychiatrists in the recent times, though it should be an integral part of assessment, and studies clearly mention that 5% of the patient of epilepsy, they commit suicide. It is 10 times higher than in general population in patients with epilepsy. And I said that very commonly, it is usually either occurring in the post-ictal period or in the interictal period. The risk factors, clear-cut, psychiatric illnesses have been identified as a universal antecedent to people committing suicide. Psychosocial circumstances are there, they are important, but are of secondary importance. As I mentioned previously, interictal dysphoria is commonly associated with episodic suicidal thoughts or moods. Temporal lobe epilepsy is an independent risk factors. And I said, as I mentioned, 25% increase in suicide with temporal lobe epilepsy. There are studies talking about anxiety disorders associated with epilepsy with a point prevalence of around 20%. It's a very, very high number. It's all about how much we are seeing the patient of epilepsy and how much actually we are inquiring into the psychiatric manifestations. People, they do present with dissociation also with epilepsy. And I must tell you, my dear friends and my seniors, it's a very, very tricky situation whenever you are getting cases of dissociative convulsions along with true seizures. And yes, it is there. It is very well a reality. And at times it becomes very difficult to differentiate what is happening with what and when. I've already talked about something called epileptic fugue or choreomania. A little bit about sexuality. Both women and men, they with epilepsy, they experience problems in sexual functioning across all phases. Men particularly have increased risk of erectile dysfunction, which might be associated with the illness or by the side effects of medications. So it can be both ways around. People, they do present with adjustment disorders, that fear that and the seizure happens again, what will happen to me in my life. Now I'm going to complete my presentation with few slides, little bit talking about treatment induced or treatment emergent psychiatric sequelae. We all know that we engage into pharmacy or polytherapies. There are dosing levels. There are concepts of reduced folate, tryptophan, or maybe family history of or a past illness of personal history of psychiatric illness. Usually, the problems associated with AED are behavioral manifestation and depression. Psychosis is very, very rare, but mind it, I'll talk about it. There is a drug which has been rampantly used both by psychiatrists and neurologists in the last 2-3 years, which is associated with increased risk of psychosis. Classical readings, they say that barbiturates, now we don't use barbiturates anymore, apart from few drugs here and there. Barbiturates, they present with hyperactivity and conduct problems. One of the largest population-based study was conducted in Bangladesh, where they found that use of phenobarbital in child and adolescent was present with increased rate of conduct problems. It can also be associated with depression, behavioral changes, and cognitive dulling, so on and so forth. Phenytoin is known to cause irritability, somnolence, psychomotor slowing, and cognitive difficulties. Carbamazepine and valproate are relatively safe, but long-standing use will present with some cognitive side effects. I'll not talk about vigabatrin or tiagabine, which are not commonly. What I was talking about is levetiracetam. There are enough evidences and studies to say that levetiracetam is associated with behavioral activation, which may present from a plethora of or the range of behaviors from catatonia to psychosis. It has been found that psychosis and mood symptoms can be seen even during the tapering or withdrawal period of various anti-epileptics. Epilepsy surgery, yes, temporal lobectomy is very commonly associated with increased risk of depression and suicide, either post-operatively just after that or maybe later stage. To summarize, whenever we are dealing with cases of epilepsy, or a psychiatrist or a mental health professional is dealing with a case of epilepsy, is always better to follow a ask or tell approach. Definitely, we have to find an optimal seizure control with an individualized treatment, but at the same time, please spend time screening for mental health issues. And mind it, there are no investigations. It is purely through direct questioning, educational programs, routine forms. If you make such, we have to identify and eliminate correctable causes, promptly treat and refer. If a person is suffering from, a psychiatric sequelae, seek help from a psychiatrist. The ultimate goal is to have freedom from seizures and an optimal quality of life and well-being. So there is definitely a relationship between epilepsy and psychiatric conditions. The comorbidities, they assume greater importance in the recent times, particularly all the levetiracetam use and development of psychosis. There are frequent associations with impaired function, quality of life, and we still have to do and work a lot in understanding of the advances in neurobiology of the pathophysiological relationship between epilepsy and psychiatry. So with this, I come to the conclusion. I end my presentation with a glimpse of the place where I work, Central Institute of Psychiatry. Thank you very much, everyone, for giving me this opportunity to spend time with you. Over to you, the chairpersons. If there are any questions, I'm happy to take. Thank you so much.
Thank you, Dr. Nishant, for such an exhaustive presentation. And I mean, you have really covered all the points. It's very important that we definitely, suppose we see a patient of epilepsy, we have to inquire about the various psychiatric aspects. We should give time to the patient. And similarly, if that patient is having epilepsy, we should consider inquiring about the various psychiatric symptoms. So with this, if there are any questions, Dr. Prajakta, in the chat box, I don't see any here.
Not yet, ma'am.
Can I add to something?
Yeah, sure, Dr. Niti.
We recently saw a patient, he is a patient with psychosis since many years, with frontoparietal lesion there and the MRI stable. And she was put on zonisamide and of course risperidone, and then they were like looking for tardive dyskinesia. They said there's a rapid perioral tremor movement, which on my examination I didn't find. It doesn't rule out. But interestingly, when I looked up, zonisamide itself gives rise to tardive dyskinesia, almost all the anti-epileptics. So we have to be very careful when we are doing this, because if we in a hurry reduce our antipsychotics and we land up with more problem because it's TD, it could be a major problem. Another thing I have seen in another patient was somebody with cerebellar issues they had, and they got admitted, and the person had frontal lobe issues in the MRI. So that got the activated symptom, because then I looked up later on that cerebellum has got an inhibitory effect on the frontal lobe. So the moment the cerebellum got involved, the inhibitory effect went away on the seizure, and the activity came up like a seizure activity, which manifested only like a behavior thing, with hallucinations. So these two things came to my mind, that when you go into this kind of consultation layers, it's very important. All our cases, and frequently we see hyponatremia in the consultation liaison. Almost all anti-epileptics, barring phenytoin, all anti-epileptics. It becomes our job to keep on regularly looking at these issues. And that's my input. A very good talk, Dr. Nishant. Thank you so much.
Similarly, I think one should really take care of these non-convulsive status epilepticus conditions, because there, and if it's a chronic, one gets to confuse with psychiatric illness per se. And if you increase the antipsychotics, many times you land patient into a lot of trouble, and the seizures also increase. Plus, sometimes I have seen patient really, deteriorating to a large extent. What's your take on this, Dr. Nishant? This non-convulsive.
Yes, ma'am, definitely. Ma'am, I'll give you an anecdote. Just few days back, maybe 1.5 months back, we lost a patient in our hospital. So the only presentation, he was diagnosed with schizophrenia, admitted in the ward and in the hospital. And somehow we missed that the aggressive episodes were very stereotypic. They would come and go. And in one of those aggressive episodes, he actually went on and stabbed himself by a sharp object, and he succumbed to the injuries. So it was just like that. And it's very simple to differentiate at times if you are mindful of all these things. A simple neurological examination just after the person gets better from that episode or aggression. As I said, it can present with anything: aggression, confusion, psychosis, irritability. The only thing which we have to look into is the stereotypic pattern. If there's something which is happening repeatedly and there is a pattern to it, always suspect that are we missing a seizure phenomenon. And it is not a bad idea to confirm it with a neurological examination, if possible to do an EEG examination. You will definitely get some ictal or interictal changes in the EEG during that time. And adding anti-epileptics might do wonders in such cases, ma'am.
Similarly, regarding the forced normalization, like I've had many patients, saying कि डॉक्टर साहब दौरा पड़ेगा तो बहुत अच्छा, आप 1 महीने तक ये शांत रहेगा. So that is also something, you know, which they have to be told that they should not leave it at that. It's a part of the illness.
Absolutely, ma'am. We see such phenomena day in and day out. So if you are handling such cases, you will come across such phenomena, ma'am. Thank you.
Dr. Prajakta, any questions from you?
Not yet, ma'am. No questions from the audience. Thank you, chairpersons. Thank you.
So over to the organizers.
Thank you so much for inviting us.
Yeah.
Thank you.
It was a pleasure, you know, learning experience, all the 3 sessions.
Yes.
Yeah.
Hello,
So,
sir.
Hi, Nilesh.
Sir, and I would request Dr. Nilesha sir to give sir final words of thanks, and then we can end the session for today.
Thank you.
Sir, unmute.
Thank you, Sagar. I think we had a wonderful session, and all the 3 lectures were very, very informative and very, very useful. Some of the theoretical questions were also important because sometimes we need to know the theory behind the practice. So I think, and the last lecture was very, very useful for the postgraduate students. So I'm very grateful to all the 3 speakers and all the chairpersons and Prajakta also for comparing. Thank you very much to everyone, and good night.
Thank you.
One comment. We just want to remind for tomorrow's session, and so this will be an in-person session tomorrow. So those in and around Mumbai can please. We encourage you to attend. It will also have 2 MMC points, and the topics are also really nice. So you can join us at 9 o'clock for the inauguration, and then the day will go ahead.
Yeah.
Wish you all the best for tomorrow's program, and thank you for inviting us.
Thank you, madam. Thank you. We had more than 200 participants today online also. So it's, I think, a great success and a great start for the midterm CME of IABP. Thank you.
Congratulations for that.
Thank you, madam. Thank you.
Okay.
Good night. Good night, sir. Good night.
Good night, everyone.